let the panel become familiar with Adobe Connect. We truly appreciate all your efforts to allow us to help navigate this app and also to become familiar with it and to try to make this meeting run as smoothly as possible. Good morning and welcome. For media and press, I would like to announce the FDA press contact is Nathan Arnold. His email is nathan.arnold@fda.hhs.gov, and his phone number is 301-796-6248. My name is Alberto Papo, and I will be chairing today's virtual meeting. I will now call the morning session of the Pediatric Oncology Subcommittee of the Oncologic Drugs Advisory Committee to order. We will start by going down the meeting roster and introducing ourselves. We will use a call-response method, and we think that this will work better. I will call the panel's member name to prompt the member to speak, and then we will ask the panel member to introduce themselves into the record. We will start with David Mitchell. We will then go to...
Dr. Papo, I'm sorry, I had to unmute myself. I'm David Mitchell. I'm a consumer representative, but more importantly, I'm a cancer patient. I have multiple myeloma and am in continuous treatment, have been for almost 10 years.
Thank you. My name is Alberto Papo. I'm the chairperson of the pedsODAC. I'm a pediatric oncologist, and I work at St. Jude. Dr. Jonathan Cheng.
Good morning, Jonathan Cheng. I'm the industry rep, and I'm with Merck Pharmaceuticals.
Dr. Catherine Bollard.
Hi, this is Dr. Catherine Bollard. I'm the director for the Center for Cancer and Immunology Research at Children's National and the George Washington University here in Washington, D.C.
Dr. Ira Dunkel.
Good morning. My name is Ira Dunkel. I'm a pediatric neuro-oncologist at Memorial Sloan Kettering Cancer Center in New York City.
Dr. Julia Glade-Bender.
Good morning. My name is Julia Glade-Bender. I'm also at the Memorial Sloan Kettering Cancer Center in New York City, where I serve as the Vice Chair for Clinical Research in the Department of Pediatrics.
Dr. Richard Gorelick.
Good morning, everybody. I'm Richard Gorelick. I am the Division Head of Pediatrics at MD Anderson Cancer Center in Houston, Texas.
Dr. Theodore Lech.
Hi, good morning. I'm Theodore Lech. I'm a pediatric oncologist at UT Southwestern Medical Center in Dallas.
Donna Lewinsky.
Good morning. Donna Lewinsky from Solving Kids Cancer in New York.
Dr. Andy Kolb.
Good morning, this is Andy Kolb. I'm the director of the Nemours Center for Cancer and Blood Disorders at Nemours Alfred I. DuPont Hospital for Children in Delaware.
Dr. Catherine Janeway.
Good morning, I'm Katie Janeway. I'm a pediatric oncologist and sarcoma expert at Dana-Farber and Boston Children's Hospital in Boston, Massachusetts.
Dr. Nainesh Kamani. Dr. Kamani, if you can hear us, can you introduce yourself for the record?
Good morning. Can you hear me?
Yes.
Hi, good morning. I'm Nainesh Kamani, pediatric immunologist and bone marrow transplanter at Children's National Hospital, Washington, D.C., and at George Washington University.
Dr. Toby McDonald.
Good morning. This is Toby McDonald. I'm Director of Pediatric Neuro-Oncology at Emory University and Children's Healthcare of Atlanta.
Dr. Leo Mascarenas.
Good morning. I'm Leo Mascarenas, the Deputy Director of the Cancer and Blood Disease Institute at Children's Hospital Los Angeles, where I also serve as the Head of Oncology.
Dr. William Parsons.
Hi, this is Will Parsons. I'm a pediatric oncologist and Deputy Director of Texas Children's Cancer and Hematology Centers at Baylor College of Medicine in Houston, Texas.
Dr. Elizabeth Retz.
Good morning. This is Elizabeth Retz. I'm a pediatric oncologist and division director at New York University.
Dr. Nita Seibel.
Hi, this is Nita Seibel. I'm a pediatric oncologist in the Clinical Investigations Branch of CTEP at the National Cancer Institute.
Dr. Malcolm Smith.
Good morning. I'm Malcolm Smith and the pediatric oncologist in the Cancer Therapy Evaluation Program at the National Cancer Institute.
Can we have more slides, or we're done with the slides? Can we have the next slide? Okay, Dr. Latoya Bonner.
Good morning. This is Latoya. I am the DSO for this meeting.
Dr. Greg Riemann.
Good morning. This is Gregory Riemann. I'm the Associate Director for Pediatric Oncology and the FDA's Oncology Center of Excellence.
Dr. Denise Casey.
Hi, good morning. This is Denise Casey. I am a pediatric oncologist and acting team lead for sarcoma melanoma at FDA.
Do we have any additional slides? I think that's pretty much, well, I think we needed Dr. Leslie Doris. Do we have a picture of her on the next slide?
Well, if we don't, hello, this is Leslie Doris. I'm a pediatric oncologist at the FDA in Division of Oncology 3.
And also Christine Lincoln is from the FDA.
Hi, I'm attending as well, and I don't have a picture.
Okay. I think that's pretty much everybody. Did I leave anybody out? And would you like to introduce yourself for the record? Okay, we will proceed then. For topics such as those being discussed at today's meeting, there are often a variety of opinions, some of which are quite strongly held. Our goal is that today's meeting will be a fair and open forum for discussion of these issues and that individuals can express their views without interruption. Thus, a gentle reminder, individuals will be allowed to speak into the record only if recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the Government in the Sunshine Act, we ask that the advisory committee members take care that their conversations about the topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with the FDA about these proceedings. However, FDA will refrain from discussing the details of this meeting with the media until its conclusion. Also, the committee is reminded to please refrain from discussing the meeting topic during breaks or lunch. Thank you. We will proceed with the FDA introductory remarks from Dr. Greg Riemann.
Good morning. I just want to also extend a welcome to the advisors and to our pharma company sponsors for this virtual meeting of the Pediatric Subcommittee of ODAC. This is a first. I appreciate your flexibility. As in the past, our focus at this meeting is to accelerate the timely development of novel anti-cancer agents with potential applicability to pediatric cancers. At present, the only legislative initiative relevant to pediatric cancer drug development is the Best Pharmaceuticals for Children Act. We will hear presentations and discuss two products in early development under investigational new drug applications in an attempt to maximize the agency's authority under BPCA, which is a voluntary program. Those products are SP2577, Cephalosporin, the epigenetic modifier from Salarius, and Marizomib, proteasome inhibitor from Bristol-Myers Squibb. Company presentations and expert panel discussions and recommendations will serve to help inform the review divisions of the Office of Oncologic Diseases and the Office of Tissue and Advanced Therapies in the Center for Biologics and the Oncology Center of Excellence as to whether written requests for pediatric assessment should be issued for these products based on the degree of unmet clinical need and the potential public health benefit to children, the extent of non-clinical data and clinical data in adults to warrant and support pediatric investigations, and a review of the benefit-risk considerations. So again, I would like to thank you for your service to the committee and your service to the agency. Thank you.
Thank you very much, Dr. Riemann. Dr. Latoya Bonner will read the conflict of interest statement for the meeting.
Good morning. The Food and Drug Administration is convening today's meeting of the Pediatric Oncology Subcommittee of the Oncologic Drug Advisory Committee under the authority of the Federal Advisory Committee Act, FACA, of 1972. With the exception of the industry representatives, all members of the committee and temporary voting members of the subcommittee are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of the subcommittee's compliance with federal ethics and conflict of interest laws covered by but not limited to those found at 18 U.S.C. Section 208 is being provided to participants in today's meeting and to the public. FDA has determined that members of the committee and temporary voting members of the subcommittee are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflicts when it is determined that the agency's need for a special government employee's services outweighs his or her potential financial conflict of interest, or when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Related to the discussions of today's meeting, members of the committee and temporary voting members of the subcommittee have been screened for potential financial conflicts of interest of their own as well as those imputed to them, including those of their spouses or minor children, and for the purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, quotas, teaching, speaking, writing, patent and royalties, and primary employment. For today's agenda, information will be presented regarding pediatric development plans for two products that are in the development for an oncology indication. The subcommittee will consider and discuss issues relating to the development of each product for pediatric use and provide guidance to facilitate the formulation of written requests for pediatric studies if appropriate. The product under consideration for this session is SP2577, presentation by Salarius Pharmaceuticals, Incorporated. This is a particular matters meeting during which specific matters related to SP2577 will be discussed. Based on the agenda for today's meeting and all financial interests reported by the committee members and temporary voting members, Conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208, B3 to Drs. Ira Dunkel, Julie Gladbender, Richard Gorlet, Theodore Leitch, and Leo Mascarenhas. Dr. Dunkel's waiver involves consulting interests with 3 companies for which he receives remuneration between $0 to $5,000 per year from 2 companies and between $10,001 and $25,000 per year from a 3rd company. Dr. Gladbender's waivers involves her employers. Contract for a study of SP2577 funded by Salarius Pharmaceuticals. Dr. Gorlick's waiver involves his employer's contract for a study of SP2577 sponsored by Salarius Pharmaceuticals and funded by the National Pediatric Cancer Foundation. Dr. Latch's waiver involves 3 of his employer's research contracts. One is funded by the Children's Oncology Group, the second is funded by the Neuromastoma and the Medullomastoma Translational Research Consortium, and the third is funded by ASI. Dr. Mascarenhas' waiver involves 2 of his employer's research contracts. One is a study of SP2577 sponsored by Salarius Pharmaceuticals and funded by the National Pediatric Cancer Foundation, and the other is a study funded by AstraZeneca. The waivers allow these individuals to participate fully in today's deliberations. FDA's reason for issuing the waivers are described in the waiver documents, which is posted on the FDA's website at www.fda.gov/advisory-committees/committees meeting materials/drugs/default.htm. Copies of the waivers may be obtained by submitting a written request to the agency's Freedom of Information Division. The address is 5630 Fishers Lane, room 1035, Rockville, Maryland, 20857, or requests may be sent via fax to 301-827-9267. For the record, Dr. Stephen Dubois has self-recused from participating in this session of the meeting. To ensure transparency, we encourage all standing committee members and temporary voting members to disclose any public statement that they have made concerning the product at issue. We respect FDA's invited industry representative. We'd like to disclose that Dr. Jonathan Chang is participating in this meeting as a non-voting industry representative, acting on behalf of regulated industry. Dr. Chang's role at this meeting is to represent industry in general and not any particular company. Dr. Chang is employed by Merck. We would like to remind members and temporary voting members that if the discussions involve any other products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all participants to advise the subcommittee of any financial relationships that they may have with the firm at issue. Thank you.
Thank you very much, Dr. Bonner. Both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages all participants, including the applicant's non-employee presenters, to advise the committee of any financial relationships that they may have with the firm at issue, such as consulting fees, travel expenses, honoraria, and interest in the applicant, including equity interests and those based upon the outcome of the meeting. Likewise, the FDA encourages you at the beginning of your presentation to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your presentation, it will not preclude you from speaking. We will now proceed with Salarius Pharmaceuticals' presentation. Are we able to start the presentation?
Hi, good morning, sir. We're working on it.
This is David Arthur, CEO of Salarius. We pre-recorded the presentation and submitted it. I believe we're looking for the committee to begin the recording.
Yes, sir, that is correct. We're working on bringing that up.
Good morning. I'm David Arthur, CEO of Salarius Pharmaceuticals, and on behalf of the entire Salarius team, I'd like to thank the committee for inviting us to review the seclidemstat development plan. We are looking forward to today's discussion. Joining me today as presenters are Dr. Bruce McCready, Chief Scientific Officer, Dr. Damon Reed, Principal Investigator of our ongoing Ewing sarcoma clinical trial, and Dr. Margaret Dugan, Senior Medical Advisor. Over the past few years, we have achieved several development milestones, including orphan drug designation, rare pediatric disease designation, IND activation, initial enrollment in both of our ongoing clinical trials, and most recently, in December of last year, Fast Track designation. These milestones all support the ongoing development of seclidemstat for the treatment of relapsed or refractory Ewing sarcoma. Why are we here? As every member of the committee knows, Ewing sarcoma is a devastating disease predominantly affecting children and young adults. Salarius is currently completing dose escalation, and at this moment in time, input and feedback from the committee could be pivotal in the development of this potential new treatment for patients that truly need new treatments the most. We believe preclinical data supports pursuing a Ewing sarcoma indication, and as mentioned, we are completing dose escalation and will then begin dose expansion by treating a larger group of patients at the maximum tolerated dose. We are currently exploring potential tumor engagement and efficacy markers. We are seeking committee feedback on how best to identify efficacy signals in our clinical program. The current study population is generally heavily pretreated with high tumor load, and unfortunately, the patients are progressing. We believe epigenetic therapies require time for epigenetic reprogramming, and we want to ensure that efficacy in this population is identified so that we can continue to quickly develop seclidemstat for these patients in need. In addition, we would also like the committee's input on innovative trial designs that support the identification of efficacy signals and support the overall seclidemstat development program. Salarius is committed to developing seclidemstat for patients in need and look forward to the committee's input on how to optimize this process. I'd like to now introduce Dr. Bruce McCready to review mechanism of action, our design rationale, and preclinical data.
Thank you, David. Epigenetic enzymes represent attractive targets for cancer therapeutics due to their role in regulation of gene expression. These enzymes can modify DNA and histone, resulting in changes to chromatin structure to a densely packed closed state, which is non-permissive for transcription, or to a relaxed open state, which is permissive for transcription. In addition, many epigenetic enzymes associate with repressive or activating protein complexes to affect regulation of gene expression. Overactivity of epigenetic enzymes can result in changes to the normal transcriptional balance and lead to cancer development and progression as a result of upregulation of genes associated with tumor growth and downregulation of tumor suppressor genes. Drugs that inhibit epigenetic enzyme activity can help treat cancer by reversing dysregulation of gene expression and restoring a normal transcriptional state. LSD1 is an epigenetic enzyme that affects gene transcription via its FAD-dependent enzymatic domain to demethylate mono and dimethyl histone 3 lysine 4 and 9 residues, thereby modifying chromatin structure and access to transcriptional machinery. In addition, LSD1 can affect repression and activation of transcription by interacting with various activating and repressive protein complexes via its tower domain. LSD1 activity is required for normal hematopoiesis, maintenance of stemness and differentiation, as well as roles in cell motility, epithelial to mesenchymal transition, and autophagy. Overexpression of LSD1 is associated with tumor genesis and disease progression as a result of both its enzymatic histone demethylase activity and interactions with various transcriptional regulatory protein complexes. High levels of tumor-associated LSD1 expression is associated with a poor prognosis. First-generation LSD1 inhibitors bind irreversibly at a site in the catalytic domain and prevent binding of the required cofactor, FAD. Although potent inhibition of LSD1 enzymatic activity is achieved, these compounds are associated with hematologic toxicity, mostly neutropenia and thrombocytopenia. In addition, first-generation irreversible inhibitors do not inhibit many of the scaffolding protein-protein interactions between LSD1 and various transcriptional co-regulatory protein complexes. Shown on the right, SP2577, seclidemstat, is a first-in-class reversible inhibitor of LSD1 that binds at a novel site within the enzymatic domain. This novel and reversible binding may explain why SP2577 demonstrates more extensive inhibition of LSD1 scaffolding protein interactions, as well as a decreased risk of hematologic toxicity. LSD1 can remove both transcriptionally permissive and repressive histone marks. Pictured in the left panel of this slide is that seclidemstat, like other LSD1 inhibitors currently in development, inhibits LSD1 demethylation of mono and dimethyl H3K9 to prevent the activation of previously silenced genes. Inhibition of LSD1 enzymatic activity by seclidemstat in the P53 prostate cancer cells results in increased repressive methyl marks on histone 3 lysine 9. However, due to its unique binding site and reversible binding, seclidemstat is also able to inhibit LSD1 scaffolding activity with DNA binding proteins and regulatory complexes such as transcriptional co-regulators that are associated with oncogenesis. Shown on the right side of the slide is seclidemstat's ability to inhibit association of LSD1 with the androgen receptor to prevent activation of androgen receptor target genes in the LNCAP prostate cancer cell line. Ewing sarcoma is driven by a fusion oncoprotein that results from chromosomal translocation between EWS and ETS gene family members, such as ERG and FLY1. In approximately 90% of cases, a T1122 translocation results in production of the EWS FLY1 fusion oncoprotein. The EWS FLY1 fusion protein is a transcription factor that interacts with coactivators and co-repressors that may also recruit LSD1 to drive the activation of tumor growth genes and deactivation of tumor suppressor genes. Ewing sarcoma cells highly express LSD1 and are dependent on LSD1 activity for survival. Seclidemstat inhibits the growth of Ewing sarcoma cells by disrupting LSD1's association with coactivators and co-repressors that act in concert with the EWS FLY1 oncoprotein to promote transcription of genes that are associated with oncogenesis or repress the expression of tumor suppressor genes. Shown on the right side of the slide is the impact of treatment of A673 Ewing sarcoma cells with SP2509. This was a first-generation compound that is structurally similar to SP2577, seclidemstat. Cells are treated with 2509, vehicle or control shRNA, or an shRNA targeting the AOS fly mRNA. As we can see from the heat map of upregulated and downregulated genes, Treatment with SP2509 results in the reversal of many up and down-regulated genes that are driven by EWS-FLI activity in much the same manner as knockdown of EWS-FLI protein levels by targeted shRNA. The inset to the right of the heat map shows that A673 cells that highly express the EWS-FLI oncoprotein are more sensitive to growth inhibition by SP2509 than are cells that show little or no expression of EWS-FLI protein. SP2577 shows anti-proliferative activity against a panel of Ewing sarcoma cell lines with IC50 values ranging between 185 and 1269 nanomolar. In the SKN-MC mouse xenograft model of Ewing sarcoma, SP2577 shows potent tumor growth inhibition that results in a significant increase in survival and complete cures in 80% of treated animals. Given Seclidemstat's proposed mechanism of action in Ewing sarcoma, additional sarcomas are of interest for future clinical trials because they either share a similar translocation to EWS-FLI1 that interacts with LSD1 and or have elevated LSD1 expression and are sensitive to LSD1 inhibition. These sarcomas that affect pediatric populations include desmoplastic small round cell tumor, which often results as a translocation with EWS-WT1, myxoid liposarcoma, which includes translocations between EWS-CHOP as well as fuse-CHOP, as well as rhabdomyosarcoma and osteosarcoma, which display an increased level of LSD1 expression and rely on LSD1 activity for proliferation and colony formation. Now I'd like to introduce Dr. Damon Reed from the Moffitt Cancer Center.
Thank you, Bruce. I'm Damon Reed. I'm an associate professor and I'm the principal investigator for the Seclidemstat phase 1 trial. Next slide. Ewing sarcoma is relatively rare amongst cancer, but very common in the pediatric age range with 400 new patients diagnosed every year, with a median age firmly in the pediatric space of 15 years of age. 3/4 of patients present with localized disease and 1/4 present with metastatic disease. All of these patients are treated with a standard of care up front of 29 weeks of chemotherapy, 35 inpatient days, and surgery or radiation for local control. These therapies can lead to cardiotoxicity, secondary cancer, and other morbidities. Next slide. There has been some improvement by intensifying therapy and adding more drugs with Ewing sarcoma over the decades, but no improvement in metastatic survival. Unfortunately, relapse disease mirrors this poor survival curve shown in metastatic Ewing sarcoma on the next slide. About 1/3 of patients will relapse of their disease, and they have a very poor outcome with less than 10% long-term survival. There are no FDA-approved agents for relapsed Ewing sarcoma, and this relapsed Ewing sarcoma is an area of unmet need, with being the 3rd most common tumor enrolled on the PD match trial, which is available for patients who don't have other clinical trial options. Next slide. So while there are relapse regimens, there is no standard of care for relapsed Ewing sarcoma. And this table shows many of the regimens that are used. There's very little prospective evidence, with much of this borrowed from single institution studies with few patients, and there's no published randomized evidence comparing these regimens. While there are responses in these relapse regimens, complete responses are very rare. Next slide. It is in this context of a poor standard of care for relapsed Ewing sarcoma and poor outcomes that led to the phase 1 trial that we're conducting with Seclidemstat as a first-in-human trial in Ewing sarcoma. I'm proud that our phase 1 trial has correlates to advance the science of this disease, as relapsed Ewing sarcoma is poorly understood. And these include cell-free DNA, looking for digital droplet PCR for the Ewing sarcoma translocation, and other novel technologies to look for a biomarker of tumor in the blood. Circulating tumor cells as well to look for Ewing sarcoma cells, and other biomarkers such as lactate dehydrogenase, pharmacodynamic biomarkers like hemoglobin F concentrations, which may rise with LSD1 inhibition, and in dose expansion, serial frozen biopsies required at screening, cycle 2, and at the end of therapy to evaluate Seclidemstat's effect and resistance. In this rare relapse population of Ewing sarcoma, a historical cohort should be considered with ongoing work to help with threshold setting. And event-free survival bars, such as presented by Dr. Angie Collier with relapsed Children's Oncology Group phase 2 studies of 12% point estimate for 6 months EFS, and the ongoing RECUR trial will add prospective evidence to this. Response rate can also be considered. And now I'd like to introduce Dr. Margaret Dugan.
Thank you, Damon. Good morning. I will be presenting an update on the early clinical program of Seclidemstat in Ewing sarcoma. The first-in-human phase 1 study is being conducted in patients with relapsed refractory Ewing sarcoma. As with all phase 1 studies, the primary objective is to evaluate the safety and tolerability of single-agent seclidemstat across multiple escalating doses administered as a 75 milligram tablet strength in the fasted state as a BID dosing regimen given daily. Secondary objectives include determination of MTD, characterization of PK, evaluation of the effect of food, and preliminary anti-tumor activity in these patients. As Damon has previously stated, exploratory objectives include assessment of cell-free DNA, circulating tumor cells, and tumor tissue for pharmacodynamic markers of disease burden, tumor response, and drug effect. Cell-free DNA will be analyzed to quantify the EWS/ETS translocations. Circulating tumor cells will be quantified and also assessed for gene expression profiles. Tumor biopsies will be assessed for genome-wide expression patterns, mutational profiles, as well as LSD1 protein levels. Next. Looking at the key study eligibility, patients must have a histologic diagnosis of Ewing sarcoma that is refractory or recurrent, including at least one prior course of therapy, which must have contained a camptothecin-based regimen, or it was contraindicated, or the patient declined such treatment. Patients are at least 12 years of age and at least 40 kilograms in weight. Patients must have a good performance status and radiographic evidence of measurable disease for dose expansion patients. This phase 1 study is enrolling onto 7 dose escalation steps, starting at 75 milligrams to 1,500 milligrams BID from 8 U.S. sites. The study began with an accelerated dose escalation design in which single patient cohorts were enrolled until a drug-related grade 2 or higher adverse event or a DLT was observed in cycle 1, at which point a classic 3+3 design started, enrolling at least 3 patient cohorts. The dose escalation will stop upon observation of 2 DLTs during the first cycle in a cohort of 3 to 6 patients, or when the 1,500 milligram BID dose level has been determined to be safe. This will define either the maximum tolerated dose or the maximum acceptable dose. Then, at that dose, a total of 20 patients will be enrolled to further define the safety and preliminary anti-tumor activity of seclidemstat. The study is currently enrolling onto the 1,200 milligram BID dose level. Of the 16 patients enrolled as of our data cutoff of December 2019, the median age was 25 years, with 88% of patients being between 18 and 68 years old. 63% were male, 69% had good performance status, 90 or higher. The majority of patients had surgery and/or radiation therapy. 81% had a prior camptothecin-containing regimen. 69% had received 3 or more prior regimens. All patients had a gene rearrangement of EWSR1 as per local assessment. Although not an entry requirement, the majority of patients had measurable disease at baseline. Sites of metastases are typical for this patient population. The median time from initial diagnosis to study drug was 4.2 years, with the majority of patients being 2 or more years from their initial diagnosis. Overall, this patient population represents a heavily pretreated group of Ewing sarcoma patients. Cycle 1 single-dose pharmacokinetics have been assessed in 13 evaluable patients treated at doses of 75 to 900 milligrams BID. Under fasting conditions, a proportional and linear increase in AUC and Cmax has been observed. The half-life is approximately 5 to 8 hours. Using PK modeling at 900 milligrams BID, exposure is expected to be above 1,000 nanograms per ml for approximately 16 to 20 hours per day, while at 1,200 milligrams BID, exposure is expected to be above that level for the full day. 1,000 nanograms per ml represents the expected efficacious concentration based on preclinical studies. In conclusion, Dose escalation continues at the highest doses to define the MTD or MAD, and at clear dose levels, seclidemstat is safe and tolerable. PK demonstrates dose proportionality with sustained exposure for up to 24 hours at these higher doses. The study population represents an advanced, heavily pretreated group of Ewing sarcoma patients with extensive disease involvement who define an unmet medical need. Seclidemstat is a novel, selective, reversible LSD1 inhibitor developed to address this unmet medical need, which selectively targets the underlying mechanism of disease to improve patient outcomes. Salarius continues its phase 1 studies with a commitment to the pediatric population and are seeking guidance on the appropriate studies for a proposed pediatric study request. This concludes our presentation. We are happy to take your questions.
Thank you very much. We will now take clarifying questions for Salarius Pharmaceuticals. Please use the raise hand icon to indicate that you have a question. Please remember to put your hand down after you have asked your question, and please remember to state your name for the record before you speak. It would be helpful to acknowledge the end of your question with a thank you and end of your follow-up question with a that is all for my questions, so we can move on to the next panel. So I see Julia Glade-Bender. Julia, would you like to ask a question?
Yes, please. Good. Bender from Memorial Sloan Kettering. Thank you very much for the presentation this morning. I was wondering two things. The first question is why was a prior camptothecin regimen required for study entry for the phase 1 trial? And then the second question is if you could please review any preclinical data that you have using cyclodemstat in combination with chemotherapy. Thank you.
Yes, this is Margaret Dugan. I'm the Senior Medical Advisor for Salarius, and I can take the first part of that question. At the time, this was a first-in-human phase 1 study for patients with Ewing sarcoma, and it was felt that they must have had failed a standard of care therapy. I'd like to ask Andriata to answer the second part of the question. Dr. Duncan?
Hi, this is Andriata Duncan, Associate Director of Non-Clinical Development. Thank you, Dr. Dugan. We do have some preclinical data in combination with chemotherapy with cyclodemstat. Those data were actually generated by Dr. Damon Reed, who is on the call, so I would like to pass this question along to him.
Okay, thank you very much. Excellent questions, both of them. Technically, on this current trial, just to answer number one, camptothecin is not required, but a discussion regarding that is required. So when we designed the trial, wanted to make sure that that patients at least knew of other therapies, and so we kind of built that into the inclusion criteria. But technically, at this moment and from the beginning of the trial, patients could have failed standard first-line therapy on Ewing sarcoma and gone directly to cyclodemstat, but so far that has not occurred. In terms of the preclinical, the second question, we have studied in vitro across multiple cell lines in Ewing sarcoma, and we do this guided by a paper from 2013 that suggests using clinically achievable doses and in presence of protein concentrations that reflect protein binding and for durations of exposure that would match the human PK. And while we didn't have that for cyclodemstat, we did test this agent along with some others. And in general, cyclodemstat both combined well with synergy or additive effects across a broad spectrum of different traditionally used therapies like SN38, the derivative of irinotecan and topotecan, or 4-HC, a cyclophosphamide derivative, or etoposide. So in general, cyclodemstat shows promising in vitro combination activity in Ewing sarcoma cell lines.
Thank you. That's all my questions.
Next is Dr. Katie Janeway.
Yes, I would like to thank the presenters for the very informative presentation. I am wondering if you are able to share any information about toxicity at this point. Thank you.
Yes, thank you. This is Margaret Dugan. So the current safety profile does not define any prohibitive toxicities, and there are no treatment-related study discontinuations or deaths. The overall frequency of treatment emergent adverse events related to seclidemstat of grade 3 or 4 is very low. And we have seen one DLT of nausea, vomiting, and abdominal pain judged to be treatment-related, and the trial continues in its dose escalation phase at the 1,200 milligram dose.
So this is Katie Janeway with just one follow-up question. What are you able to share
or otherwise might help us to discern what is the most powerful biomarker that could be used for patient selection of those most likely to respond?
Yes, hi, this is Margaret Dugan. So I think in terms of the biomarkers we are exploring in the clinic, we are looking at circulating tumor cells for gene expression profiles during the dose escalation to look for any expression patterns that could solidify exactly the inhibition of LSD1 by our agent. I think more importantly is, and we're close to this, when we get to the disease expansion, we will be able to have those pre and post tumor biopsies and be able to explore to a better extent those changes in gene expression profiles, demonstrating that we are hitting the target and effectively translating that into tumor responses. The other part of your question, I'm sorry.
No,
yeah.
I guess it's just a general comment. When I look at the LSD1 expression, for example, in rhabdomyosarcoma, and it's not clear to me whether those were alveolar or embryonal models, there is the level of expression of LSD1, but that may or may not communicate how dependent the tumor is on LSD1 in terms of what is driving its growth. And the question is really whether that is determined by a translocation or whether LSD1 expression in and of itself, a high level of it, is actually predictive of response.
Right, and we will, during this extensive biomarker, be looking at the different EWSR1 translocation patterns, and also essentially by IHC measure LSD1 protein level expression.
Okay, the next question is from Dr. Malcolm Smith.
Yes, this is Malcolm Smith, and I think the first point I wanted to make was with the increased protein expression in the absence of a genomic alteration, and this is not predictive of clinical benefit for targeted agents in most settings. And so in the absence of functional genomic studies, it's hard to know what role LSD1 inhibition and or knockdown might have in terms of therapeutic potential for other pediatric solid tumors. And I would endorse Alberto Papo's comment about the PPTC data as well. A specific comment about the immune checkpoint inhibitors. Ewing sarcoma, as one example, is an immunologically cold tumor for which there's really no evidence of response to checkpoint inhibitors, and really little or no evidence for the immune system being able to recognize these tumors as foreign. And so before getting into combinations with an agent like SB2577 in a cold tumor like Ewing sarcoma, I think it would be really important to see preclinical models of immunologically cold tumors with low tumor mutational burden and or the ability of an agent like SB2577 to convert an adult immunologically cold tumor into, with low tumor mutational burden into a checkpoint responsive tumor.
Thank you, Malcolm. The next question is from Dr. Gorlick.
It's Richard Gorlick. I find some of the most compelling preclinical data, the evidence around the reversal of the transcriptional signal driven by EWS5. So I am somewhat, intrigued about the idea of the spectrum of translocations associated with EWS that may show activity in response to this. I think that can be probed in the context of clinical trials by getting the break apart fusion, so you know whether it's specifically EWS-Y or a different binding partner. The other areas that you could think about exploring are desmoplastic small ram cell tumor, which has already come up earlier. There's not a lot of preclinical models, but a couple do exist, and obviously it's a clinical entity as well. I also wouldn't forget the adult sarcomas. So clear cell sarcoma also has an EWS fusion. And I wonder whether you have data thus far or are planning to obtain data to explore the spectrum of EWS, related activity. Thank you.
Margaret Dugan. I guess, Dr. Groliker, you're asking Salarius to answer this, correct?
Yeah, I'm questioning whether you have any data on clear cell sarcoma, desmoplastic small round cell tumor beyond what's already been mentioned, or plans to look at those.
Right. So thank you. You refer back to my statements about in the clinic how we've advantageously been able to enroll some of these patients. I'd like to ask Dr. Andrietta Duncan to answer that about the preclinical activity that we may or may not have ongoing.
Thank you, Dr. Dugan. This is Andrietta. Dr. Gorlick, to answer your question, just the data that we mentioned, that one publication with DSRCT with 2509, and then the really early preclinical in vivo study that we have in clear cell sarcoma with the EWS ATF1 fusion, those are the extent of the data that we have at the moment. But certainly, I do agree with you that understanding the biology with different fusions is certainly warranted and something that we could consider doing.
Thank you.
Thank you.
This is Dr. McCready. I'd like to also respond to your question that we are, in fact, evaluating over 160 different cell lines in a very extensive study right now where we will be asking some of the very questions you are, including looking for and correlating specific mutations in various chromatin remodeling complexes. We have identified some that we know do sensitize more to our inhibitor, such as the SMARCA4 and the ARID1 that are part of the SWE/SNP pathway. We are also continuing with our combinations for immuno-oncology, and that your question about a cold tumor such as a Ewing's tumor making it hot. We have looked at tumors including CO434, which is normally an immunologically cold tumor, and have noted that we do turn that into quote-unquote a hot tumor, that we can demonstrate clear-cut infiltration now by mononuclear cells, by T cells into those tumors after incubation with the compound. We've also seen it in the NCF7 line. So I think your comments are right on target, and rest assured we are very thoroughly evaluating all of these different questions to try and ascertain better different potential combinations, mechanisms, as well as what are the specific targets, as you saw from the RNA-seq profile. Can we identify specific other targets, which we believe are more likely in the case of Ewing's not to be the EWS protein itself, but rather one of its many interacting co-repressor or co-activator complexes. One of the proteins within those complexes is more likely a target.
Okay, and then Dr. Cheng, and I would like to remind the panel that the purpose of this session is really to have discussion amongst panel members. So we will have Dr. Cheng ask the question.
Sure, thank you, Dr. Papo. So I actually had a question regarding kind of the context of this question of other tumors. My question is actually to the FDA, if they can give guidance as to how they think about how a compound should be investigated in other tumors. The extent of the investigation, should it be limited based on the science, knowing that this is difficult to investigate every single tumor, particularly in the context of a written request. I do think that it would be helpful to have an understanding as to how the FDA is viewing the opportunities or the limitations in investigating other tumors and how extensive or limited it should be.
I don't know if Greg wants to answer that or another member of the FDA.
And this is Greg Riemann. I can try to answer Dr. Cheng's question. part of the reason for this question was because of the fact our policy, if you will, in issuing written requests is to make sure as best we can that the investigational drug that's being explored addresses as many or all of the potential public health considerations in the pediatric age group. So when we consider issuing written requests, we want to make sure that that we are addressing all of the possible indications that might be addressed by a particular drug in children. So in saying that, we're not requiring that sponsors necessarily do exhaustive investigation of every single pediatric cancer in this situation. But the question here was raised because of the lack of clarity regarding LSD1 expression versus the existence of one or more specific fusions related to the various partners that was associated with the proliferative activity and the growth of Ewing's, and then also with the activity of SP2577. So that was the basis for the question. I've answered it for you.
Thank you, Dr. I forgot to identify myself as John Chang, industry rep, and I do appreciate a practical approach, particularly for sometimes small companies or even big companies wish to be focused in their investigation of other tumors, and sometimes preclinical or early clinical data is sometimes thought to be maybe adequate as to how to investigate this. But thank you for the helpful response.
Sure. And if Dr. McCready could please lower his hand. So if there are no additional questions, I will try to summarize what we discussed for question number one. The first one was that it is unclear whether increased protein expression of LSD1 will predict any kind of response, and additional studies, including functional genomics, are highly encouraged. In addition to that, the preclinical data that was presented for other tumor types seemed, in my opinion, a little bit weak, especially for alveolar rhabdomyosarcoma and osteosarcoma, so perhaps additional studies need to be conducted. There also was talk about combination therapy with LSD1. However, we were unable to see that data, so it would be important to see if there's really a synergistic effect by adding other chemotherapeutic agents or other therapies to LSD1 inhibitors. There was an issue about considering brain tumors for this specific drug. If I understood correctly, that blood barrier penetration is relatively low. I think it was 3%. So I am not sure that additional studies in brain tumors should be conducted or not, but I'll ask our CNS experts to chime into that portion of the summary. There was also a suggestion to further explore potential biomarkers that will allow to a better prediction of response in this patients. There was also suggestion that a combination with immune therapies is to be conducted, that additional preclinical studies are done, since Ewing sarcoma specifically appears to be a cold tumor. There was also a question about increasing the spectrum of when to use this therapy in other EWS rearranged tumors, including desmoplastic small round cell tumor and clear cell sarcoma. And finally, Dr. Riemann explained the FDA's view when considering a written request and to explore all the possible indications of a particular drug. Could you raise your hand or say if I missed anything? Specifically, Toby, did I address the CNS issue correctly, or did I miss something?
Sorry, this is Toby. No, I think you addressed it correctly. The question would be whether 3%, the activity of the drug, is viable as a mechanism at that level. We know that other chemotherapy such as cisplatin has a very low percentage across the blood-brain barrier, about 2 to 4%. So it really comes down to activity of the drug at that level.
Thank you very much. If I didn't mess up very badly or if I didn't miss anything, we're going to move to the next question. So the FDA would read the second question to the committee.
I'm just waiting for the slide to change. Oh, question 2. Thank you. So for discussion point number 2, given the non-clinical results of synergistic effect and increased anti-tumor activity of SP2577 in combination with chemotherapeutic and epigenetic agents and immune checkpoint inhibitors, consider its use as a combination treatment in pediatric tumors.
If there are no questions or comments concerning the wording of the question, we will now open the question for discussion. Dr. Katie Janeway has a question.
I'm sorry, that was to raise my hand for discussion.
Anyone else wants to ask? I think some of these points were discussed in the question number 1. I think Dr. Greg Riemann has his hand raised.
Yeah, I was just going to say the same thing, Alberto. I think we actually covered many of these points in our discussion of the previous question.
Let me just keep going down the list and see if there's anything else that we are missing. Malcolm? Malcolm, do you have a question?
Alberto, I guess the question, are we having discussion now on this point or questions about this point?
No, we're having discussion about this point.
Okay, well then, and so Katie will join in as well then. But the point I would make here is that the best predictor for success in combination regimens is activity as a single agent, and not every single agent with activity will improve outcome when it's used in combination, but agents without single agent activity have a much lower likelihood of improving outcome when used in combination. You know, to illustrate this, we did a retrospective study looking at CTEP-sponsored randomized phase 2 trials in which an experimental agent was added to a known active agent. And fewer than 3% of these randomized trials that involved experimental agents without documented single-agent activity for the disease under evaluation produced results that were indicative of likely true clinical benefit. And so I think, you know, the appropriate step now would be to look for the single-agent activity against Ewing sarcoma like Salarius is doing. And we're certainly all hoping that there's going to be a good, robust activity observed. And at that point, it's a no-brainer to proceed to combination studies. If there's not single-agent activity, then I think we'll have to think long and hard about what the next steps are for Ewing sarcoma or other, you know, EWS rearranged tumors. And that would have to be done in part based on what preclinical data that existed, but also in the context of other research opportunities for patients with Ewing sarcoma. Thank you.
Thank you very much. Katie still has her hand raised. Do you want to have a little bit of discussion on this question?
Yes, I wanted to comment on this question. So thank you for asking this question. I think it's a very important question, which is why it came up in the discussion of the first question. So I do think that if there is evidence of single agent activity, particularly if the toxicity signal remains reassuring, that there will need to be consideration of combination studies with chemotherapy. If you are thinking about the sort of long-term, more long-term in terms of in which setting would you imagine that this drug would be used in Ewing sarcoma, most likely you would want to study this in the newly diagnosed setting. And in that setting, you would likely be combining it with some type of Ewing sarcoma-directed chemotherapy. Even if you were to, set your ultimate goal on use of this, for example, in first recurrence, it's very likely that you would want to combine this with one of the chemotherapy regimens that is used at the time of relapse in Ewing sarcoma. So I would encourage the company and the other investigators who are working with this compound to continue to study in the preclinical space activity in combination with chemotherapy agents that are used in Ewing sarcoma. I do also think that it would be very interesting to better understand the mechanisms by which there might be synergy with immune checkpoint inhibitors, as was already discussed, in terms of converting what is thought to be a cold tumor into a more hot tumor in the setting of combination with immune checkpoint inhibitors. Thank you very much.
Thank you, Katie. Leo, you're next.
Hi, this is Leo Mascarenas. Can you hear me?
Yes, we can.
This question is an intriguing one, and when you look at sort of pediatric sarcomas in particular, they're rapidly proliferating and aggressive cancers, and oftentimes the time to progression, especially at the time of relapse, is relatively low. So I struggle with, if there's a lot of robust preclinical activity, testing of a drug in a phase 1 setting to make decisions based on just purely on clinical activity in that setting may be challenging. And I think the example which we all know well was, I think, with temsirolimus and rhabdomyosarcoma, mTOR inhibition, where single agent therapy didn't show any exciting clinical activity, but the preclinical information was very strong, and the combinatorial therapy in preclinical models was also excellent, and we were able to show at least some activity in the relapse setting, and is now being tested in the upfront setting. While single agent activity would be very encouraging and would help us to move this rapidly forward, if there is robust clinical activity, preclinical activity, I'm not sure whether we absolutely need clinical activity in the phase 1 setting to think of a possibility of moving a very active agent preclinically forward. Though it will be helpful, clinical activity will be helpful, but I don't know if it should be the sole decision.
Even in the presence of the fact that all of the patients that are being tested with these drugs are patients with Ewing sarcoma, so if you didn't see any responses, and I think it's 16 or 17 patients, despite the fact that you have robust preclinical activity, you would still consider moving this forward with combination therapy?
So if the combinatorial, if there's preclinical combination data which can be generated, which is robust, particularly in Ewing sarcoma, and we have a hint of clinical activity in terms of some disease stabilization rather than responses, given the landscape of potential active agents in Ewing sarcoma, I might consider at least a trial in the relapse setting.
Thank you very much. I see Katie still has her hand up. Do you still want to comment on this question, or you're okay, Katie?
Apologies, I'll raise my hand.
So if there are no additional comments on this question. If I could briefly summarize this, a lot of the issues raised in this question were already addressed in question number 1. However, one point of view would be to look for single agent activity in this clinical trial, and if there is no robust single agent activity, it would be difficult to justify combination studies. On the other hand, there is a precedent for preclinical studies that have shown activity of some single agents, for example, temsirolimus, and lack significant clinical activity as a single agent. However, when they were combined with chemotherapy in the preclinical models, they appeared to be a synergy, and actually in the clinical setting, they proved to be efficacious in the setting of relapsed rhabdomyosarcoma. So one point of view would be, if there is no single agent activity, try not to pursue combination studies. The other point of view from the panel is if there is very strong preclinical activity with combination therapies, to at least pursue a phase 1 clinical study in the relapse setting of Ewing sarcoma. Did I capture the conversation okay, or did I miss anything, or did I mess up anything? Leo or Malcolm?
That was clear, Alberto, from my perspective. This is Malcolm Smith.
Okay, thank you very much.
My perspective too.
Okay, thank you very much. We will now proceed to the third question. The FDA will read the third question.
Hi, for our last discussion question today, please discuss the use of SP2577 in patients less than 12 years of age, given the range of tumor types that appear to be susceptible to the anti-tumor effects of SP2577 based on non-clinical data.
We will now proceed with the, if there are no questions or comments concerning the wording of the question, we will now open the question for discussion. I see Leo has his hand up. Leo?
As a pediatrician, I would support investigating this in patients less than 12 years of age, at least in select cancers. The median age of Ewing sarcoma is in the teenage years, and we do have several patients who are below the age of 12 who could potentially benefit.
Thank you very much. I also see I see a bunch of hands. The next one is Dr. Gorlick.
It's Richard Gorlick. I'm actually going to recommend the other way. Ewing sarcoma, even though there are patients who are below 12, the vast majority are above it. Clear cell sarcoma is an adult condition, and desmoplastic small round cell tumor is an adolescent and above disease. Most of the entities you're talking about are going to be easily feasible for an activity study to be done in an over 12 group. And although you ultimately, if there's activity, may want to know the activity in younger the safety in younger patients, you're going to be able to get an activity assessment in the older age range. And I'm not sure there's enough histologies to justify needing to do a PEDS 12 and below study right away. Thank you.
Elizabeth?
Hi, this is Elizabeth Rates. I think one thing, if it were to be considered in the less than 12-year-olds, would be to have a better understanding of plans for alternate formulations. It may be hard in a pill form that's a small pill, so it's a fair number of pills to take with each dose. So it might be helpful to understand if other formulations are planned.
Thank you. Katie?
Yes, I was going to say very much something similar to what Richard Gorlick said. I think that you can wait to expand to understand dosing and tolerability in patients under 12 years old with Ewing sarcoma until you have signals from your dose expansion in terms of whether or not there's evidence of clinical activity.
Thank you. And just a brief reminder to please introduce yourself when you're commenting on the question, and then go ahead. And I see that hands are still raised for Elizabeth, for Katie, and for Leo. Unless you have another question, please lower your hand. And let me see where we're at here. I have Leo still.
You hear me?
Ted? Yes, we can hear you. Go ahead.
This is Leo Mascarenhas. I was on mute. For testing in children younger than 12 years of age can take a while, and waiting for activity in Ewing sarcoma may hamper its further development in pediatrics, particularly if preclinical data could be used to generate information to test this in patients with rhabdomyosarcoma, where the majority of patients are less than 12 years of age. So considering it broadly, issues related to pharmacokinetics, safety, as well as formulation is important in my opinion. Thank you.
Thank you very much. Ted is the next one.
Hi, Ted Leitch. I was just going to second what the other advisory committee members have said, that while we certainly can look at activity in patients over 12, it will take some time to think about a pediatric formulation if one is necessary, or at least smaller dosing increments like your current dosing increment.
One. He knows what he's doing.
That would enable a pediatric study, and I would encourage Salarius to think about those issues earlier rather than later, so that it isn't only after there's evidence of activity in adults that those begin to be considered.
Thank you, Ted. Dr. Cheng? Okay, Ira?
Ira Dunkel, Memorial Sloan Kettering. I wanted to ask fellow committee members who are more sarcoma-oriented than I am if we should be surprised that there have been so few adolescents who've been enrolled on the existing trial. I realize that's a little tangential to this question, but might impact on even younger patients choosing to access the drug. Thank you.
I think part of it would depend on the number of centers that have this clinical trial open and the type of patients they're seeing, if they're mostly adult centers. I mean, I don't have that clinical trial open here, so I will ask some of the panel members that have their clinical trial open at their institution if it's an issue of older age of the patients with relapse Ewing's, or the fact that they're mostly seen by your adult counterparts. I think I see Katie's hand, so Katie, go ahead.
So I would say it's a combination of both of the factors. Oh, sorry, Katie J, my pediatric oncology, Dana-Farber. I would say it's a combination of both of the factors you mentioned, Alberto. One is the sites where this trial is open, and the other is actually the age of our relapsed Ewing sarcoma patient population, who tend to be older. So even when we run a trial like this through the Children's Oncology Group, which is exclusively pediatric centers, we see a good quarter of the patients being over age 18.
And Ted has a comment also.
Apologies, I just didn't put my hand down.
Okay, are there any additional comments about this question? Leo has one. Yes.
I was just going to add that, I mean, the outcomes relatively for Ewing sarcoma have improved in recent years, and younger patients tend to have a better prognosis. The other requirements is that oftentimes there are other therapies which are usually considered at first relapse, and so many of the patients, as you could see from the data which was presented, were beyond first relapse, and that may contribute also to the older age of the patient.
Thank you, Leo. So if I could summarize the discussion on this third question, most of the panel members that commented on this question feel that we should have more data on patients over 12 years of age because the vast majority of patients that present with Ewing sarcoma will be in this age group. However, some panel members think that specifically for other histologies other than Ewing's, for example, rhabdomyosarcoma, if there are clinical trials that are going to be conducted in this specific histologies, that age less than 12 years of age should be taken into consideration for enrollment to the clinical trial. In addition to that, that would give us the opportunity to better assess the PK of this compound in this population and also investigate alternative formulations. Regarding the rate of enrollment of younger patients in the current ongoing trial, the reason why there's such few patients might be related to the fact that some of this, the number of institutions that have this clinical trial open, the age of patients with Ewing sarcoma at the time of relapse, the fact that some of these patients have improved outcomes and go on other therapies prior to going to experimental therapies. For example, the RECUR trial is the perfect example, irinotecan, temozolomide, ifosfamide, cyclotopo. So I think I've summarized the discussion of question number 3. Unless there's anything I've missed, I welcome your comments or suggestions. Okay, if there are no additional comments, we will now break for lunch. We will reconvene at 1:20 p.m. Eastern Standard Time. Panel members, please remember that there should be no discussion of the meeting topics during lunch amongst yourselves or with any member of the audience. Thank you very much, and we will see you back in a little bit. Thank you very much.
Hi, panel members. We would like for you to stay in the meeting room in Adobe Connect. You may hang up and use your phone during lunch, and please remember to call back in by 1:05 so we can make sure we have you connected audio for the afternoon session. Thank you.
Audio
We will begin the afternoon session in 2 minutes.
Oh, Jesus.
Audio recording for this meeting has begun.
Okay, good afternoon. Can you hear me okay?
Yes, we can.
Okay, good afternoon and welcome back. For media and press, I would like to announce the FDA press contact is Nathan Arnold. His email is nathan.arnold@fda.hhs.gov, and his phone number is 301-796-6248. My name is Alberto Papo, and I will be chairing today's virtual meeting. I will now call the afternoon session of the Pediatric Oncology Subcommittee of the Oncology Drugs Advisory Committee to order. Like we did in the morning, we will use a call-response method where I will call the panel's member's name to prompt the member to speak, and then the panel member will identify, introduce himself or herself, and then we can put that into the record. So we will start with Alberto Papo. I'm the chairperson of the Pediatric ODAC. I'm a pediatric oncologist at St. Jude Children's Research Hospital. Dr. Jonathan Cheng.
Good afternoon, Jonathan Chang. I'm the industry rep, and I'm with Merck.
Dr. Catherine Bullard.
Hi, it's Dr. Catherine Bullard. I'm from Children's National and the George Washington University in Washington, D.C.
Dr. Stephen Dubois. Steve, if you can unmute your phone.
Hi, I'm here now. Steve Dubois, pediatric oncologist from Dana-Farber Boston Children's in Boston.
Dr. Ira Dunkel.
Hi, this is Ira Dunkel, a pediatric neuro-oncologist at the Memorial Sloan Kettering Cancer Center in New York.
Dr. Julia Glade-Bender.
Hi, I'm Julia Glade-Bender, also from Memorial Sloan Kettering in New York City. I'm a pediatric oncologist and vice chair for clinical research in the Department of Pediatrics.
Dr. Richard Gorlick.
I'm Richard Gorlick. I'm the division head of pediatrics at MD Anderson Cancer Center in Houston, Texas.
Dr. Theodore Leitch.
Leitch, a pediatric oncologist at UT Southwestern Medical Center in Dallas, Texas.
Donna Lewinsky.
Donna Ludwinski, Solving Kids Cancer in New York.
Dr. Andy Kolb. Andy, are you on the line? We will come back to him. Dr. Katie Janeway.
Dr. Katie Janeway, a pediatric oncology and sarcoma specialist at Dana-Farber and Boston Children's in Boston, Massachusetts.
Dr. Nainish Kamani.
Good afternoon. This is Nainish Kamani, pediatric immunologist, bone marrow transplanter at Children's National in Washington, D.C.
Dr. Toby McDonald.
Good afternoon. This is Toby McDonald, pediatric neuro-oncologist at Emory University and Children's Healthcare of Atlanta.
Dr. Leo Mascarenas.
Hi, this is Leo Mascarenas. I'm the Deputy Director for the Children's Cancer and Blood Disease Institute at Children's Hospital, Los Angeles, and the head of oncology and a pediatric oncologist.
Dr. William Parsons. Okay, I will try to call their names at the end of this. Next slide. Dr. Elizabeth Wright.
Hi, Elizabeth Rates, pediatric oncologist and division director at New York University.
Dr. Nina Seibel.
Hi, Nita Seibel, pediatric oncologist in the clinical investigations branch at CTEP at the National Cancer Institute.
Dr. Malcolm Smith.
Hi, Malcolm Smith in the Cancer Therapy Evaluation Program at the National Cancer Institute.
Dr. Latoya Bonner.
Hi, good afternoon. This is Latoya Bonner, DFO for this meeting.
Dr. Gregory Riemann. Okay. Dr. Denise Casey.
Hi, this is Denise Casey, pediatric oncologist, Division of Oncology 3, FDA.
Do we have another slide? Dr. Leslie Doris. Dr. Megan Zimmerman.
Hi, this is Megan Zimmerman. I'm a pediatric oncologist and a clinical reviewer in the clinical hematology branch at FDA.
Okay, so before we go any further, I just want to call out a couple of names that we're missing. Dr. Kolb, are you on the line? Dr. Parsons.
Yes, I'm here. Sorry about that, Alberto. Will Parsons from Texas Children's Hospital and Baylor College of Medicine in Houston, Texas.
Thank you. Dr. Doris. And Dr. Riemann, which I hear he's in the participant pod, but he'll be moved up to the presenters pod. Okay, so we will get started. For topics such as those being discussed at today's meeting, there are often a variety of opinions, some of which are quite strongly held. Our goal is that today's meeting will be a fair and open forum for discussion of these issues and that individuals can express their views without interruption. Thus, a gentle reminder, individuals will be allowed to speak into the record only if recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the Government in the Sunshine Act, we ask that the advisory committee members take care that their conversations about the topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with the FDA about these proceedings. However, the FDA will refrain from discussing the details of this meeting with the media until its conclusion. Also, the committee is reminded to please refrain from discussing the meeting topic during breaks or lunch. Thank you. Dr. Latoya Bonner will read the conflict of interest statement for the meeting.
Thank you, sir. The Food and Drug Administration is convening today's meeting of the Pediatric Oncology Subcommittee of the Oncologic Drug Advisory Committee under the authority of the Federal Advisory Committee Act, FACA of 1972. With the exception of the industry representatives, all members of the committee and temporary voting members of the subcommittee are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of the subcommittee's compliance with federal ethics and conflict of interest laws covered by but not limited to those found at 18 U.S.C. Section 208 is being provided to participants in today's meeting and to the public. FDA has determined that members of the committee and temporary voting members of the subcommittee are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflicts when it is determined that the agency's need for a special government employee's services outweighs his or her potential financial conflict of interest, or when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Related to the discussions of today's meeting, members of the committee and temporary voting members of the subcommittee have been screened for potential financial conflict of interest of their own as well as those imputed to them, including those of their spouses or minor children, and for purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, quotas, teaching, speaking, writing, patents, and royalties, and primary employment. For today's agenda, information will be presented regarding pediatric development plans for 2 products that are in development for an oncology indication. The subcommittee will consider and discuss issues relating to the development of each product for pediatric use and provide guidance to facilitate the formulation of written requests for pediatric studies if appropriate. The product under consideration for this session is Marizomib, presentation by Celgene International II Sàrl, a wholly owned subsidiary of Bristol-Myers Squibb. This is a particular matters meeting during which specific matters related to Marizomib would be discussed. Based on the agenda for today's meeting and all financial interests related by the committee members and temporary voting members, conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208, B3 to Drs. Ira Dunkel, Theodore Leitch, and Leo Mascarenhas. Dr. Dunkel's waiver involves consulting interests with 4 companies for which he receives remuneration between $0 and $5,000 per year from 3 companies and between $10,001 and $25,000 per year from a 4th company. In addition, his employer has a contract for a study funded by Bristol-Myers Squibb. Lastly, Dr. Dunkel serves as the chair of the Pediatric Brain Tumor Consortium, which funds studies and receives support for studies from Apexagen, Pfizer, Celgene, Novartis, Lila Soli, and Novacur. Dr. Leitch's waivers involve 2 of the employees' research contracts. One is funded by the Children's Oncology Group, and the second is funded by Novartis. Dr. Mascarenhas' waivers involve his employer's contract for a study funded by AstraZeneca. The waivers allow these individuals to participate fully in today's deliberations. FDA's reason for issuing the waivers are described in the waivers document, which are posted on FDA's website at www.fda.gov/advisory-committees/committees meeting materials/drugs/default.htm. Copies of the waivers may also be obtained by submitting a written request to the agency's Freedom of Information Division at 5630 Fishers Lane, Room 13, room 1035, Rockville, Maryland, 20857. Or requests may be sent via fax to 301-827-9267. For the record, Dr. David Mitchell has been recused from participating in this session of the meeting. To ensure transparency, we encourage all standing committee members and temporary voting members to disclose any public statements that they may have concerning the product at issue. With respect to FDA's invited industry representative, we would like to disclose that Dr. Jonathan Chang is participating in this meeting as a non-voting industry representative, acting on behalf of regulated industry. Dr. Chang's role at this meeting is to represent industry in general and not any particular company. Dr. Chang is employed by Merck & Company. We would like to remind members and temporary voting members that if the discussion involves any other products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all participants to advise the subcommittee of any financial relationships that they may have with the firm at issue. Thank you.
Thank you very much. I see that Dr. Kolb just joined. Andy, do you mind just stating your name for the record and introduce yourself?
Yeah, hi, this is Andy Kolb. Sorry, I was a little late getting back.
Thank you. Both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the advisory committee meeting, the FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages all participants, including the applicant's non-employee presenters, to advise the committee of any financial relationships that they may have with the firm at issue, such as consulting fees, travel expenses, honoraria, and interest in the applicant, including equity interests and those based upon the outcome of the meeting. Likewise, the FDA encourages you at the beginning of your presentation to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your presentation, it will not preclude you from speaking. We will now proceed with Celgene International II Sàrl's presentation.
Hello, Dr. Papos. Can you hear me?
Yes, we can hear you very well.
Great, thank you. So good afternoon. I'm Debbie Toddy. I'm a Global Regulatory Strategy Lead for Oncology at Celgene, a Bristol-Myers Squibb company. On behalf of BMS and the Marizomib team, I would like to thank the FDA and Dr. Gregory Riemann and his team for the invitation to participate in today's Pediatric ODAC meeting. We are here today to present information and to gain feedback on Marizomib, a proteasome inhibitor that we are developing for the potential treatment of children with high-grade gliomas, including glioblastoma and diffuse intrinsic pontine glioma. During our presentation today, we will present information on the topics shown on this slide. First, Sherry Leonard, our Global Regulatory Lead for Marizomib, will share our commitment to working collaboratively to advance pediatric research. She will provide a summary of the overview of the Marizomib development strategy and the regulatory history, and she will highlight key activities related to pediatric development. Next, Dr. Mark Kieran, our clinical lead in pediatric oncology, will review the Marizomib mechanism of action. He will provide a summary of the phase 1/2 clinical trial experience in adults, and he will walk us through the ongoing and planned clinical trials for children. For the Q&A session, I will serve as the moderator and will direct the questions from the advisory committee to one of our Marizomib team subject matter experts to respond. I would now like to turn this presentation over to Sherry Leonard.
Thank you, Debbie. Before I get started, I just want to ask if anyone can see the slides.
We can see the slides.
I cannot see the slides, and I think there's others that have said the same
I
thing.
cannot see the slides as well, so I wanted to make sure before I start that they are visible to most people.
They're not visible.
While we do this, Greg, do you mind introducing yourself?
Yes, sorry. I was waiting to be automatically connected as the instructions were, but unfortunately I had to sign off and get back on, so I apologize for being late. But I'm Gregory Riemann, Associate Director for Pediatric Oncology in the Oncology Center of Excellence. I apologize for the technical snafu here and inability to see the slides.
Can everybody see the slides now, or we're still working on it?
All I see is a green circle twirling around.
Others of us can see them.
Good afternoon. This is Yvette Waples from the FDA. Can we please put a five-minute pause while we try to figure out the connection?
Absolutely.
Thank you.
Can everybody see the slides now?
Yeah.
Yes, they're visible now.
Good afternoon. If you still cannot see the slides, you may consider to disconnect your VPN. What we realize is a lot of people, when they have VPN connected, it occupies some bandwidth and cause delay in internet.
It looks like most people that were not able to see the slides are now able to see them. Greg, can you see them and everybody else can see them?
Yes, I can see them. Fine.
Okay, let's proceed with the presentation then.
Thank you. This is Sherry Leonard, Global Regulatory Lead for Marizomib. Celgene and BMS are committed to pediatric cancer research. This includes early evaluation of the oncology pipeline using the molecular target to identify potential pediatric tumor types, as well as early discussions with FDA for alignment on pediatric development plans. Our goal is to improve treatment options for children with cancer by decreasing the lag time between adult and pediatric studies and obtaining written requests earlier. As covered in Dr. Riemann's introduction, we are here today to gain advice on the potential role of Marizomib in the treatment of pediatric cancers and on the optimal design of studies that may serve as part of a written request. Today we will present an overview of the Marizomib program, including development plans for pediatric patients with high-grade glioma, including GBM and DIPG. The adult Marizomib development program is focused on newly diagnosed GBM based on two key findings from the phase 1-2 studies. First, Marizomib is an irreversible proteasome inhibitor that crosses the blood-brain barrier. This is evident from findings in non-clinical studies and the observed dose-related CNS AEs in adult clinical studies. And second, these CNS AEs, while reversible, determined that the overall benefit-risk profile for Marizomib was more favorable for use in patients with CNS tumors. The phase 1-2 clinical studies have led to a phase 3 study being conducted by the EORTC that is adding Marizomib to the standard treatment of temozolomide and radiation therapy followed by temozolomide. The pediatric development strategy for Marizomib is based on the molecular target of proteasome inhibition and CNS penetration, and therefore is focused on high-grade glioma, including GBM and DIPG. We are here today to gain advice for a potential written request focused on high-grade glioma, including GBM and DIPG. On slide 6, you will find the regulatory history and key activities for pediatric development. In 2006, the first adult studies began in advanced solid tumors, lymphoma, and multiple myeloma. Then in 2015, development was refocused on GBM based on non-clinical data and the observance of CNS AEs that indicated brain penetration. Beginning in 2017 and over the last 3 years, we have engaged pediatric advisors at an advisory board convened by Celgene, gained regulatory scientific advice from 3 national health authorities in the EU, and reached agreement on a pediatric investigation plan with the European Medicines Agency and an initial pediatric study plan with FDA. Both plans will be discussed further during this presentation. Pediatric expert opinion and health authority advice was sought to guide the Marizomib pediatric development plan. Based on activity observed with Marizomib and panobinostat in cell lines and models, a pediatric advisory board recommended a gated approach, starting first with DIPG, and if supported by non-clinical studies and the DIPG phase 1 study, proceeding with other pediatric high-grade gliomas. This approach was encouraged in the scientific advice received from 3 national health authorities. As described on the earlier slide, we have reached agreement with FDA and EMA on separate pediatric plans. The key difference between these 2 plans is that the FDA-agreed IPSP only includes high-grade glioma, including GBM, which was based on the adult indication, while the EMA PIP includes these studies and studies for DIPG. Celgene was encouraged by FDA to seek a written request that could include the planned studies as well as any other pediatric indications for which Marizomib could offer potential benefit. Next, Dr. Mark Kieran will present the Marizomib mechanism of action, a summary of the phase 1-2 clinical experience in adults, as well as the ongoing and planned pediatric clinical trial.
Thanks, Sherry. I'm Mark Kieran, Senior Director of Pediatric Oncology at Celgene BMS. And as mentioned previously, Marizomib is a potent, irreversible proteasome inhibitor. Proteasomes are important cellular structures that degrade unwanted or aberrant proteins within the cell. Some of these proteins may function to maintain normal cellular homeostasis,
while the TP53 protein and tumor cells benefit by removing proteins such as this. Proteasome inhibition can alter the relative contribution of proteins within a cell, including those deleterious to the tumor. In general, proteins targeted for destruction are ubiquitinated and then pass through the proteasome's central core. 3 important proteolytic enzymes, labeled beta-1, beta-2, and beta-5 within the inner core of the proteasome, digest the protein into smaller peptides for recycling. Marizomib has a lipophilic structure that allows it to pass through the blood-brain barrier and covalently binds to these 3 enzymes within the proteasome core, resulting in inhibition of protein degradation and preservation of key proteins that may reset normal cellular homeostasis. Marizomib has been studied in 5 different non-GBM phase 1-2 single-arm studies involving 280 adult patients with a variety of advanced solid tumors, lymphoma, and multiple myeloma. While the drug had manageable toxicities, a number of dose-related and reversible CNS AE events were observed. First, this suggests that the drug penetrates the brain and is supported by non-clinical data. Second, there are other proteasome inhibitors, 3 of which are approved for multiple myeloma or mantle cell lymphoma, as well as others that are in clinical trials in non-CNS diseases, such as the treatment of solid tumors, lymphoma, and hematologic malignancies. Therefore, the blood-brain barrier penetrating qualities of Marizomib has focused its development on studies in adult patients with GBM. There are 2 phase 1-2 studies of Marizomib in adults with GBM. Study Marizomib 108, which has been completed, and study Marizomib 112 that still has subjects on treatment. Study 108 had 3 parts, looking at Marizomib alone or in combination with bevacizumab, a VEGF inhibitor commonly used in adult GBM. Study 112 had 2 treatment arms and an Optune cohort. Arm 1 evaluated Marizomib in combination with radiation therapy and temozolomide, the standard of care for adult GBM, while Arm 2 evaluated Marizomib in combination with adjuvant temozolomide. The Optune cohort evaluated Marizomib in combination with temozolomide and tumor-treating fields, or TTFs, which are low-intensity electric fields. Whether as a single agent or in combination, the recommended phase 2 dose of Marizomib was 0.8 milligrams per meter squared, typically administered once a week for 3 weeks of each 4-week cycle. Study 108 assessed the activity of both single-agent Marizomib as well as Marizomib in combination with bevacizumab in recurrent GBM, as mentioned before. Some single-agent activity, as defined by radiologic responses or prolonged stable disease, was observed. See the middle column of the table. The column on the right shows the combination of Marizomib with bevacizumab, which had an overall response rate of 34% and a duration of response of 5.2 months. To help benchmark these results, a recent report of the GLOBE study of bevacizumab in recurrent GBM, which used the same RANO criteria for disease response, demonstrated an objective response rate of only 21.9% and a duration of response of only 2.2 months. While this was of interest, and recognizing the limitations of cross-study comparisons, there was no improvement in median PSF or OS, and the development of Marizomib was refined to focus on newly diagnosed GBM. As mentioned previously, the overall safety profile of Marizomib in patients with GBM, both relapsed and newly diagnosed, was similar to that observed in patients with non-GBM tumors. The most common TEAEs are listed on the slide for patients receiving Marizomib monotherapy, shown in the purple bars, Marizomib in combination with bevacizumab, shown in the gray bars, and those receiving Marizomib in combination with radiation and temozolomide, shown in the black bars.
Sorry to
Grade
interrupt
one?
you, Mark, but we cannot see those bars in this slide.
Do we want to, is there a possibility, can you go to the next slide? Okay, no, so both of them are missing the material. I could kind of point it with my hands if we were sitting in front of each other, but unfortunately, without the images here, it's going to be hard to show people the results. Alberto, do you want me to just keep going to describe what they would look like, or do you want to wait and see if those slides can be fixed?
Whatever you think. I mean, if you want to just describe the percentage of toxicities, that would be perfectly fine, Mark.
Okay. What you should have seen on slide number 13 was that the majority of the toxicities observed were grade 1 and 2 events that were frequent but not of significant grade. There were a small number of grade 3 and grade 4 adverse events that were presented there originally on the slide. These occurred only in a smaller fraction of patients. Obviously, it depended a little bit depending on whether it was monotherapy in combination with bevacizumab or in combination with radiation and temozolomide. But again, typically less than about 10% or so of patients. The most common AEs were manageable, and the vast majority, as I said, were really grade 1 or grade 2. Slide 14 was meant to show you the CNS adverse events of Marizomib, again, alone or in combination, since, as was pointed out previously, these are often the issues that led to the development of this drug in malignant gliomas. The CNS AEs that you unfortunately can't see on the slide, again, certainly supports the concept that there's blood-brain barrier penetration clinically for Marizomib. And this tended to occur early in the first dose, sometime during cycle 1 or early in cycle 2. Although you can't see the toxicities here, the two that were most problematic for patients were hallucinations, predominantly visual, as well as ataxia. Again, the CNS adverse events were reversible and resolved with dose delays, reductions, or discontinuation in medical management. Again, the majority of these toxicities were grade 1 and grade 2. I also want to point out that in the GBM program, there were 6 grade 5 TEAEs reported by clinical investigators, including 3 intracranial hemorrhage, 2 progressive disease, and 1 sudden death. None of these events, however, were considered related to Marizomib. So in summary, the safety profile of Marizomib is manageable alone and in combination with bevacizumab or radiation and temozolomide in adult GBM. Let's go to the next slide and see. Good. So in summary, although you didn't see all of the specific data, the non-clinical data and clinical experience supports the penetration of Marizomib across the blood-brain barrier. The occurrence of dose-related reversible CNS adverse events due to the blood-brain barrier penetration has shown that the risk-to-benefit ratio is more favorable for use in patients with brain tumors, where Marizomib has a manageable safety profile, as well as with preliminary evidence of activity in GBM and further potential when used in combination. Based on these results, the EORTC is conducting a randomized phase 3 trial of Marizomib, radiation, and temozolomide versus radiation and temozolomide, assessing overall survival benefit in newly diagnosed adult GBM patients. Based on the molecular target of Marizomib, its blood-brain barrier penetration, safety, and tolerability, from the adult phase 1-2 studies and the non-clinical findings that I'll detail in a moment, further development was initiated in children with DIPG. A phase 1 study of Marizomib and panobinostat in pediatric DIPG has been initiated, and additional studies in pediatric high-grade glioma are being planned. The initial pediatric clinical and non-clinical data will pave the way for future development. On the next slide is the Marizomib agreed PIP, which includes 8 different elements which are detailed in the table. In addition, 4 of these studies will also support the requirements of the FDA agreed IPSP. The quality study is focused on the development of a lower dose vial for children and will be gated based on the clinical phase 1/2 study results. The non-clinical studies assess the activity of Marizomib in combination with a number of different inhibitors in DIPG cell lines and patient-derived material, and similar work is planned for the high-grade gliomas. Identifying other more active combinations with Marizomib continues to be an important initiative. The last 3 are clinical studies, including the phase 1 safety and tolerability assessment of Marizomib alone and in combination with panobinostat in children with DIPG. Based on these results of the initial trial, a non-clinical study identifying optimal combination approaches as it becomes available in conjunction with the safety and tolerability from the phase 1 DIPG study, a phase 1/2 trial of Marizomib in combination with another drug or modality would be developed for pediatric high-grade glioma. Taken together, Celgene BMS is committed to assessing the quality, the non-clinical, and the clinical activity of Marizomib in children with DIPG and other high-grade gliomas. On this slide, which fortunately you can see, the top half is labeled Part A and shows the in vitro activity of Marizomib, panobinostat, or both in a series of pediatric DIPG cell lines as measured by relative cell viability. The blue bars are for panobinostat activity, the red bars are for Marizomib activity, and the purple bars are for the combination of panobinostat and Marizomib. In looking at the top graph on the far left, while Marizomib, again shown as the red bars, had limited activity, and panobinostat, the blue bars, had limited activity in the cell line second to the left, the 2 drugs together showed significant synergistic activity in all 6 DIPG cell lines, shown as the purple bars. In the bottom half of the slide, Part B, in vivo orthotopic assessment demonstrated activity of both Marizomib and panobinostat in a DIPG model. As mentioned previously, the phase 1 trial of Marizomib and Marizomib plus panobinostat was developed based on the non-clinical studies just presented and was designed to assess the safety and tolerability, PK, and preliminary activity of Marizomib and panobinostat in pediatric patients with DIPG. The first cycle will assess the safety, tolerability, and PK of Marizomib alone. Beginning with cycle 2, panobinostat will be added, and ongoing safety, tolerability, PK, and preliminary response will continue to be evaluated. This study has just been activated. So in conclusion, based on the manageable safety profile in adults, the CNS penetration capacity of Marizomib, preliminary evidence of activity in adult GBM, and non-clinical evidence of synergistic activity in DIPG cell lines, Marizomib is now being evaluated in children with DIPG. Celgene and BMS would like guidance for a written request that would include both patient populations, high-grade glioma as well as DIPG, and we look forward to gaining your feedback. This final slide, let me thank you on behalf of the Marizomib team, and now turn the presentation over to Debbie, who will serve as the moderator for the Q&A session.
Thank you, Mark. So for the Q&A session, as Mark said, I will serve as the moderator and direct your questions to one of our Marizomib team subject matter experts to provide a response.
Thank you very much for your presentation. We will now take clarifying questions for Celgene International. Please use the raise hand icon to indicate that you have questions. Please remember to put your hand down after you have asked your questions. And please remember to state your name for the record before you speak. It would also be helpful to acknowledge the end of your question with a thank you, and end of your follow-up question with a that's all of my questions for now, so we can move on to the next panel member. So we will start with any questions? Nita?
Yes. Nita Seibel from the NCI. So thank you for that nice presentation. And I have two main questions. First of all, can you expand more on the CNS Adverse Event Management Plan? You mentioned dose delays, interruptions, and you did mention the pattern somewhat. But how often do they recur? And then the use of antipsychotics. You said they were mainly visual hallucinations, but perhaps you could go into more detail about that. And then my second question is, particularly if you would look in the preclinical setting at rhabdoid tumors or the SMARCB1 deficient cancers. But I know with rhabdoid tumors, I think there's been a report of association with those with MYC responding somewhat to proteasome inhibitors. So if you have any preclinical data about that as well.
Thank you, Dr. Seibel. So I want to just make sure that we understand. So first, your question is, you'd like for us to expand and provide more information on the CNS management plan and how often the hallucinations recur such that you get a sense of the characterization of the events. And number two, whether we have preclinical data on rhabdoid tumors.
Yes.
So go ahead.
Yes, that's correct. Particularly to give us an idea of how this would be manifested in children or what we would expect in younger patients.
Okay. To begin with, I'm going to ask our clinical development lead for the adult programs to provide some information on the CNS management plan and how often those hallucinations recurred. That would be Dr. Ileana Elias. And then I'll redirect the second question to another responder once we've answered your question. So Ileana, are you on mute?
No, I'm here. I hope I'm not on mute. Can you hear me?
We can hear you.
So I'm Ileana Elias. I'm the clinical development physician for the adult program. We have seen the CNS-associated adverse events in about 40% of the patients, and as mentioned before, the majority, one of the hallmarks of these CNS adverse events were the visual hallucinations. In general, these occurred by cycle 2, but they could have occurred as early as after the first dose of cycle 1. The management was mainly with dose delays and dose reductions. We did not introduce any prophylaxis for these hallucinations because it was not possible to determine which patients would be most likely to experience the hallucinations. And in general, after a dose reduction, these hallucinations did not reoccur with reintroduction of treatment.
Thank you, Dr. Elias. So Dr. Seibel, did we answer the first question completely?
Yes.
Okay, and your second question was about whether we have any preclinical data on rhabdoid tumors. The short answer is we don't currently have any data on that. I can just open it up and ask if Dr. Suman Makanani or if Dr. Mark Kieran want to make any additional comments about that in terms of the thinking around obtaining additional data?
Yes, thanks, Debbie. This is Mark Kieran, the Senior Director for Pediatric Oncology. With respect to the rhabdoid tumors, or ATRTs for those in the brain, there has been a recent report, and in fact, we're in discussions with academic centers who are specifically interested in those tumors. One of the things we are interested in is identifying combination agents, so that kind of any clinical study could be gated not just on the development of this non-clinical and preclinical data, but it would also then give us a chance to incorporate the data that we obtained from the phase 1 DIPG study, vis-a-vis the dosing, the PK, the tolerability, and early evidence of activity.
Thank you, Dr. Kieran. So Dr. Siebel, did we fully answer all of your questions?
Yes, thank you.
You're welcome.
Next is Ira Dunkel.
Thank you, Alberto. Ira Dunkel, Memorial Sloan Kettering. I have 2 or 3 questions. One question kind of related to Dr. Seibel's question is, I believe that I've heard at least one presentation from a non-Celgene BMS investigator suggesting that Marizomib may have some potential for medulloblastoma. So I'd like to hear from Dr. Kieran or others if there are data about that and what the development plan might be for medulloblastoma. My second question is maybe a 2-part second question. I'm wondering if the phase 2 planned study described in the PIP depends, A, on the results of the phase 1 studies, or whether it's committed to move ahead regardless of whether there's any suggestion of efficacy in phase 1. And similarly, is it dependent at all on the ongoing ERTC phase 3 study, and when will the results from that ERTC study be available? Thank you.
Thank you, Dr. Dunkel. So first of all, we'll address the first question. Does Marizomib have some potential for activity in medulloblastoma? Do we have any data? And if so, what would be the development plan? So for this question, I'd like to direct to Dr. Mark Kieran for a response.
Yes, thank you. Ira, a great question. Yes, there is some, I think we've probably heard the same presentation vis-a-vis the potential for proteasome inhibition in medulloblastoma in early preclinical models, and we're actually in discussions with academic centers interested specifically in that disease and the role of Marizomib. Again, similar to with regard to the ATRT or rhabdoid, it would be important not just as a single agent but in combination to get as much preclinical data as possible, and then gate that along with what we learn both from that preclinical data, the preclinical data from our other tumor types, as well as the results, PK, toxicity, and evidence of activity of the DIPG trial in terms of determining the optimal way to take that information forward.
Dr. Dunkel, does that answer your question?
Yes, it did. Thank you.
Okay, Steve. I do know you had additional questions. I'm sorry.
I'm sorry, there's additional questions. I apologize. Go ahead, Ira.
Oh, I'm sorry.
Right.
I thought I posed the second question already. Okay.
You did, Dr. Dunkel. That's my that was my mix-up. So let me then direct. Your second question was in 2 parts. You were asking if the phase 2 study that was planned as part of the pediatric investigational plan would depend on the results of the phase 1 DIPG study, or if it would proceed regardless. And then the second part of that second question was whether or not the work the phase 2 study would be dependent on the results of the EORTC phase 3 study in adults with newly diagnosed GBM. And so for at least for the first part of the question with regard to whether the phase 2 study would proceed, I'm going to ask Dr. Mark Kieran to begin that response.
Great. So again, although the first study is a phase 1 study to determine the optimal dose and schedule of Marizomib in this patient population, it includes an expansion cohort. So by the end of the phase 1, there should be a sufficient signal to give an indication to justify the phase 2. So, and this is an important distinction from strictly a dose-finding study where there's very limited ability to interpret clinical activity. Here there would be an opportunity, and the trial includes both patients that are newly diagnosed after having received radiation, as well as those with recurrent disease. So I think the opportunity to develop that signal to guide the development of the phase 2 would be available at the time that decision is made. And I'll let Debbie take over vis-a-vis the EORTC question.
Thank you, Mark. So Dr. Dunkel, with regard to whether the phase 2 study would be dependent on EORTC, the phase 3 study in patients with newly diagnosed GBM, the answer is it is not gated to the phase 3 study. I think you also were interested in maybe understanding a little bit of the timing around the EORTC phase 3 study. Did I remember that correctly?
Yes.
Okay, so for the response to that, an update on the status of the EORTC phase 3 study, I'm going to ask our clinical lead, Dr. Ileana Elias, to give some information just in terms of an update on where that study is.
Yes, this is Ileana Elias. So the EORTC study is currently accruing. This study has a overall survival as a primary analysis, and we are looking at 488 events. Based on the current number of events and the projections on how these events occur, we are expecting that the final analysis will be available in the fourth quarter of 2022.
Thank you very much for answering my questions. I apologize to my colleagues if I've taken up too much of the time.
No problem. Thank you, Dr. Dunkel.
Steve, you're next.
Steve Du Bois from Dana-Farber, Boston Children's. A few questions. With other proteasome inhibitors, there's been a safety signal of peripheral neuropathy, but I didn't hear that mentioned, so I'm curious about rates of peripheral neuropathy, just thinking about potential combination partners. And then what is known about the role of the proteasome during CNS development and in pediatric patients? Might there be an impact on neurodevelopment based on what is known about this pathway in CNS development? And then the third question is just the lower age of eligibility for the EORTC trial.
Okay, Dr. Du Bois, thank you. So your first question about the observation with other proteasome inhibitors of peripheral neuropathy and whether that has been seen in the adult studies with Marizomib, I'd like to ask Dr. Ileana Elias to please respond.
Yes, so the rate of peripheral neuropathy in our phase 1 studies was extremely low. We had just about 3% of grade 3 neuropathy in one of the studies, and which was in recurrent GBM. In the newly diagnosed phase 2 GBM study, we didn't see grade 3 peripheral neuropathy.
Thank you.
Thank you, Ileana. And then, Ileana, while you're speaking, could you please address Dr. Dubois' question about the lower age of eligibility for the EURTC phase 3 study?
The EURTC study enrolls patients 18 years and older.
Thank you very
Thank
much.
you.
And then, Dr. Dubois, with your second question, you were really asking what we know about the role of the proteasome in terms of development and neurodevelopment, and how that might impact
That's exactly
children.
right. That's exactly
Okay.
right.
Okay, and so what I'd like to do is ask our translational medicine lead, Dr. Suman Makanani, if you could please address Dr. Dubois' question.
Excellent. With regard to specific role of proteasome, there are no specific targets known, although there is speculation that there could be downregulation of cell cycle and tumor suppressor proteins. With regards to what we know from a neurodevelopment standpoint as it relates to our non-clinical studies, what we can state is that from a 9-cycle, 9-month cynomolgus monkey study, Marizomib-related microscopic findings in the brain were present in male and female monkeys at the high dose, 0.45, 0.60 milligrams per meter squared, and consisted of minimal to mild axonal myelin degeneration, which was restricted to the deep cerebellar white matter. Following a recovery period of 3 months, there were no Marizomib-related microscopic findings in the brains of recovery animals, suggesting reversibility. The clinical relevance of these cerebellar findings is unknown, but may include reversible CNS AEs such as ataxia.
Thank you.
Did we
Alberto,
answer-
I don't have further questions. Thank you.
Thank you. Malcolm, you're next.
Thank you. Malcolm Smith, NCI. So most of my questions have been answered already. I did have one question about another one about the CNS adverse events, and the slide was stated that they were generally reversible. And were there any CNS AEs that were not reversible or that reversed very slowly? And then the second question related to the combination, and the combination of HDAC inhibitors with proteasome inhibitors has been studied preclinically in most adult cancers, and data very similar to what was presented for DIPG has been developed. Aside from the myeloma and the panobenostat-bortezomib combination, is there evidence for an HDAC inhibitor, proteasome inhibitor combination showing positive results in a kind of proof of concept randomized study?
Okay, thank you, Dr. Smith. So your first question is around what we observed in the adult studies for CNS adverse events. Were they, you said, we said they were generally reversible, and your question was, were any not reversible, or did they reverse slowly? So to respond to that question, I'd like to turn this over to Dr. Ileana Elias.
So we observed that the CNS toxicities did resolve upon dose reductions, and if they didn't resolve upon those reductions, once the drug was discontinued, then there were no lasting CNS toxicities. Now, this is to say that all these studies, of course, the 2 studies that we've conducted were non-randomized studies, and having these patients with glioblastoma, at times it's not that easy to differentiate what is due to the disease and the drug, but we have not seen long-lasting CNS toxicities that were thought to be associated with the drug.
Okay, thank you.
And then I think your second question, Dr. Smith, had to do with the combination of HDAC inhibitors and proteasome inhibitors. I think your point was that even though it's been studied in most cancers, aside from maybe multiple myeloma, you were asking if there's any data from a randomized study with this combination. Is that?
Right. Is there any proof of concept for, for the idea that an HDAC inhibitor, proteasome inhibitor combination actually works in the clinic the way, the in vitro synergy would suggest it might?
Okay. I think for the answer to that question, what I'd like to do is ask Dr. Kieran if you could respond to Dr. Smith's question.
Malcolm, it's, I think for me at least, a hard question to answer, so I will call on some of my colleagues, because I think the question was based on the adult data for the combination of HDAC and proteasomes, where were similar in vitro and in vivo preclinical data showed similar activity to what we showed in DIPG. Did any of those turn into activity in adult tumors? Obviously, there's no pediatric data yet, since the pediatric trial is just now beginning. I'll let others with more expertise in the adult area answer that, but one of the things is obviously the biology of tumors we know is different, and although there are general overlap mechanisms, understanding why it is that proteasome inhibitors, for example, work so well in a particular disease like multiple myeloma, but not others, means that we don't always or can't always in pediatrics rely on the adult data of different disease pathways as a perfect guide for activity in pediatrics. I know that's not a complete answer in the sense of, because we don't have any pediatric data yet. I don't know, Debbie, if perhaps Suman has any knowledge of how to answer this question.
Yeah, maybe so. Suman, Dr. Makanani, is there any additional information that you can share to respond?
Definitely. I am not aware of any proof of concept data generated in adults using HDAC inhibitor and proteasome inhibitor combination for the indications under discussion.
Thank you very much. Dr. Smith, did we answer your questions to the best of our ability?
Fine. Yeah, that's fine. Thank you.
Okay.
We only have 5 minutes left. We only have 4 minutes left for questions, so we're going to try to get as many people as possible. Toby, you're next.
Hi, Toby McDonald, Emory University. Sorry, I had a series of questions, but I'll try to be quick if possible. Just want to know if there is based on the mechanism, a biologic rationale for focus on DIPG versus other tumors. And in that setting, is there any dependency of activity on H3K27M? I saw on the slide there was wild type versus K27M, but didn't see clearly enough the difference there. And of course, that's important since adult GBMs infrequently harbor that mutation. Third, I just wanted to know whether there were data, what the rationale for panobinostat in combination versus other drugs, or particularly radiation therapy, and whether there'd be consideration of an upfront DIPG with radiation in the drug if there were data to support that. And finally, are there any biomarkers that are predictive of response or pharmacodynamic assays that can be used to track and follow whether there is a functional activity of the drug when given to the patients? Sorry for many questions.
No, it's quite all right. I was just trying to make sure that that I have all of what you'd like to know. So first of all, I think you're asking that based on the mechanism of action, is there a biologic rationale for the use in pediatric GBM? Is that correct? Dr. McDonald? I think you're on mute.
Toby, can you answer the question?
Sorry about that. Specifically DIPG in relation to H3K27M and GBM, which could have diffuse midline glioma versus non-diffuse midline glioma.
Okay, so for your first question, I think this might be a shared response, but I'm going to ask Dr. Suman Makanani if you can start based on the mechanism of action, and then perhaps Mark, you can also contribute.
Thank you, Debbie. What we know in terms of the mechanism of action, the role in terms of biological rationale, it's still largely unknown with regards to proteasome inhibition, in particular DIPG settings. What I'd like to maybe defer to my colleague Mark Kieran, if there's any additional data on this front, at least from the proteasome standpoint, we have not identified a clear biological rationale.
Yeah, thanks, Suman. Thanks, Toby. As was pointed out, we don't know exactly which proteins are being inhibited of degradation that account for the effect. But as you noted in the figure, the cell line on the very right-hand side was an H3K27 wild type, and it had the same synergistic activity as the 5 cell lines with either H3.1 or H3.3 mutations, suggesting that this is not restricted to the H3 mutational status. You're right that DIPG and diffuse midline glioma, the trial was originally defined more on the classic historical definition of DIPG vis-a-vis short symptomatology and radiographic appearance as opposed to the new molecular one. But as we talked about, particularly in the context of thinking about the development of a high-grade glioma trial, which would be kind of high grades other than DIPG and diffuse midline, it is again because, at least in the early preclinical experience, this does not seem to be restricted to H3 K27. Does that
Dr. McDonald, does that answer your first question?
That does.
Okay, and then I know you had asked for some information around the rationale for the combination with panavinostat versus other drugs or possibly radiation therapy. I'm also going to ask Dr. Kieran if he could speak to this question about how panavinostat was selected for that phase 1 DIPG study.
Yeah, thanks. Toby, in the paper that was published by the Stanford group, which was a large collaboration unto itself, they had screened literally thousands of combinations, and in the 6 DIPG cell lines, panavinostat and Marizomib was by and far the strongest signal they detected, and was again present in all 6 out of 6 cell lines. And it was based on that this kind of went forward. There was some activity, for example, when they looked at panavinostat in combination with some other pathway inhibitors, but not present in all of the cell lines and not to the same degree. And that's why this one has kind of taken precedent in terms of its clinical development. So that's how we got there. Lin et al. Is the first author of that publication from Michel Manger's lab where that data was provided.
That's helpful. Any preclinical data with radiation in suggesting upfront trial?
Debbie, is it okay if I go ahead and...
Yes, please do, Mark.
So actually as was already mentioned in terms of the adult EORTC trial, in which it will be upfront with radiation and temozolomide versus radiation and temozolomide. So some of that data will hopefully guide us in the future as we kind of see what happens there. In addition, for the DIPG study, although we will not be combining it with radiation, so as had been mentioned in the presentation, patients will get 1 month of Marizomib alone, then start the combination of Marizomib and panobinostat. But patients eligible for the trial are those that are newly diagnosed that have completed radiation, so they won't be on radiation when they start the drugs, but they will have just completed their radiation, which I know isn't quite the same thing as synergism with radiation. I'll ask Suna to comment if he knows better. I'm not aware that that was one of the combinations that they did at the Stanford study, but part of our goal is to see whether the two drugs are tolerated in pediatric patients and whether there's any signal of activity. But absolutely the idea that hopefully one could at some point, if there is activity and it's tolerable, move this more immediately up into the upfront situation, particularly for a disease like DIPG, where there really are no survivors, would be an absolutely reasonable thing to do. But step 1 and 2 is single agent and combination tolerability and signals of activity. Hopefully that answers the question.
Thank you.
I'm going to ask Dr. Riemann to, I'm just going to let, I'm sorry, we need to move on. I'm just going to let Dr. Riemann ask the last question.
Sure, absolutely.
Just briefly, and again to go back to the treatment emergent neurotoxicity. And the only intervention that I understand that was utilized was dose modification or discontinuation of Marizomib. Was that a complete discontinuation of Marizomib, or were patients allowed to restart therapy? And if so, did they have recurrent hallucinations upon restarting?
Thank you, Dr. Riemann. For your response, I'd like to ask Dr. Ilial and Elias to please comment on whether there was complete resolution following discontinuation, or if Marizomib was restarted and the CNS AEs recurred.
The recommendation was for the drug to be first dose reduced. 2 dose reductions has been allowed, and the majority of the patients, once they were dose reduced, the hallucinations did not reoccur. In fact, top of my head, I cannot recall any of the patients in our phase 1-2 studies who have had several episodes of hallucinations after they had dose reductions. But of course, for the ongoing phase 3 study where the patient population will be much larger, we don't have that data available yet.
Okay, thank you.
Does that answer your question?
It does, yes, thanks.
Okay, if you still have your hand up, please lower it. There is no open public hearing session, so we will now proceed with the charge and questions to the subcommittee and panel discussions. After each question is read, we will pause for any questions or comments concerning its wording. Then we will open the questions for discussions. So we will ask the FDA to read the first question.
Hi, this is Denise Casey. Can you hear me?
Yes, we can.
Hi, good afternoon. Thank you, Celgene, for your presentation and for the thoughtful discussion during the clarifying questions. I think a lot of these topics have been at least partially addressed during that discussion, but we will just go through them. The first question is to the committee is to please discuss your thoughts on trial design and rational combination partners for Marizomib investigation in pediatric patients with high-grade glioma.
If there are no questions or comments concerning the wording or the question, we will now open the questions for discussion. And we see Steve.
Steve DuBois, Dana-Farber. I think during the presentation I was a little bit worried that the first-in-child study would assign children to Marizomib monotherapy and keep them there, and understanding that the population of interest is rather ill, I was a little bit worried about that and was pleased to see that after the first cycle, children move on to the combination. So it's not a question, it's really a comment and maybe praise. So that was good to see. And second, in terms of rational combination partners, I seem to recall that this panel discussed ONC201 in midline glioma in the past, and I wondered if that has been looked at preclinically, since that appears to be another agent of interest in high-grade midline gliomas.
Does anybody in the panel have any insights to that question? Toby?
Sorry, I don't have any specific insights with regard to ONC201, but it relates back to another question of mine, which was trying to determine a biomarker that may be predictive of response. I don't know if any sort of sequencing analyses have been done post-drug treatment of the DIPG to understand what pathways are being affected most, which would obviously inform us better of what combinations from a functional standpoint may be the most relevant to look at, as opposed to just blasting with drug screenings, which are also important, but the complementary approach would be helpful. So as far as I know from the mechanism of action of ONC201, which is both a DRD2 antagonist and a ClpP agonist in the mitochondrial system, that there is not a specific overlap that I'm aware of, but I could be wrong.
Any thoughts on some epigenetic modifiers combined with this, given the histone mutations that you see in some of these gliomas in children? Any thoughts from the panel, Toby or Ira, or the experts in CNS tumors?
Again, I'm not specifically that I can see the connection. I would need to see exactly the pathway that is being modified.
Ira?
Thank you, Alberto. Ira Dunkel, Memorial Sloan Kettering. I wanted not to comment on that epigenetic question you just posed, but to reflect on Steve's comment about the strategy for sequentially adding the second drug after a brief window of single agent drug. And although I also agree that it's such a desperate population that this is attractive, and I am really sort of playing devil's advocate more than disagreeing, but I wonder, when you study it this way, when you then determine the tolerance of the combination, whether you can only conclude that a patient who can tolerate single agent Marizomib can then tolerate the combination at the dose, rather than that combination data being relevant for a recommended phase 2 dose for a treatment-naive population.
It's a valid point. We have Malcolm.
Yes, Malcolm Smith, NCI. Thank you, Alberto. I think, staying with the combination question or discussion, I think, obviously we all hope that the study that's ongoing with the panabinostat and Marizomib shows a good activity signal. think it will be very important, though, to understand whether the activity is more related to Marizomib and whether panobinostat actually has a contribution. As per my question, the HDAC inhibitor, proteasome inhibitor combinations have been studied preclinically across many adult cancers, but as far as I know, this has not translated to the clinic aside from the panobinostat added to bortezomib and dexamethasone for myeloma. But outside of myeloma, not aware of any translation. Also, the CNS penetration is low for panobinostat, and so I hope that if the study is a success, that we're not locked in necessarily to the combination without understanding whether single-agent Marizomib might actually be the primary activity driver.
Thank you. We have Julia.
Yes, thank you for that really interesting presentation. I was just wondering, and I may know the answer myself, but there's been a precedent in the Children's Oncology group, particularly for DIPG, to combine the new agent concurrently with radiotherapy in newly diagnosed patients. I'm wondering if this was considered as a strategy and whether it was really this concern over interpreting the CNS toxicity that is the reason that the radiotherapy would, you would initiate the drug after the radiotherapy, or whether there's a biologic reason.
Thank you. And I have Greg.
Thanks, Alberto. Greg Riemann. I just wanted to go back to what I thought was maybe a rhetorical question that Ira raised in response to Steve's statement about moving to a combination after a single cycle of single agent. And I think most patients who end up on investigational drug therapies are desperate. And irrespective of their desperation, I think we have a responsibility to learn from those patients. And I would be a little bit concerned about moving too quickly to a combination that may result in toxicity, which is unacceptable, and then abandoning a strategy and potentially abandoning a single agent that hasn't been adequately evaluated, that may take more than a single cycle before we see a signal of activity. So just a word of caution.
I don't see any other additional comments to this question. If anybody else wants to add anything, or I can try to summarize the comments for question number 1.
Alberto, this is Leo. Could I ask a question?
Of course.
Please comment.
Of course.
Leo Mascarenas from Children's Hospital, Los Angeles. I didn't get to ask this question earlier, but based on Greg's comment, I also would think a little bit of caution with combination therapy because the other JOX class effect seems to be cardiac toxicity and also is a consideration in combination with panobinostat. And while CNS tumor patients tend not to have cardiac toxicity in general because of pre-exposures, that's something to keep in mind with combination therapy.
Thank you very much. So the panel initially expressed some concerns in starting just with monotherapy, but some of the panel members were very happy to see that after the first cycle there was going to be a combination therapy. However, we strongly caution them to proceed with caution in this specific area because we do not want to abandon a potential strategy of a drug that could be potentially valuable for this disease and then stopping because of significant toxicity and some other concerns of toxicity, for example, as Leo just raised, might be cardiac toxicity. As far as which agents to combine this with, there were not a whole lot of ideas. Somebody mentioned ON201, somebody mentioned epigenetic modifiers, but there was really not a lot of other ideas. There was a comment regarding the tolerance of the combination and whether this will be enough to give us enough information for a recommended phase 2 dose. And another one of the comments was whether it has been at least contemplated to add radiotherapy with this drug, like it has been done in other trials in DIPGs in COG. I will be more than happy to add anything you want me to add, or if I missed anything, please let me know before we proceed to the next question. Everybody seems to be happy, so we will now have the FDA read the second question to the committee.
Hi, this is Denise Casey again. Again, this question alluded to in the prior discussion, but maybe we can ask the panel. Considering the CNS toxicity profile associated with Marizomib in the adult clinical experience to date, can you discuss possible risk mitigation provisions that could be included in pediatric clinical trials? And comment on any developmental or age-related assessments and management guidelines that could potentially mitigate risk in younger children who may experience CNS adverse reactions that have been relatively common and occasionally dose-limiting in the adult program.
If there are no questions or comments concerning the wording of the question, we will now open this question for discussion. So I see a hand with Toby and a hand with Leo. Toby, do you want to start?
Sorry, I was raising early for discussion.
Steve?
This is Steve Du Bois from Dana-Farber. I think the struggle here is that the target patient population with advanced DIPG, unfortunately, I mean, unless this is an active drug or an active combination, in the initial studies, it will be very challenging to follow these patients for long enough to understand at least the neurodevelopmental effects, if any, of the agent. And so I think certainly there'll be a plan to manage acute toxicities, but I think any evaluation of longer-term neurodevelopmental outcomes will likely need to come on, follow-on trials.
Thank you, Steve. Toby, do you have a comment or about this question?
Yeah, in my mind, I think this is really the most critical question, that there be very specific guidelines in place for how to mitigate with a 40 to 50% hallucination / ataxia and other CNS side effects. Life is short for these patients. Quality of life is even shorter. This is going to be in a particularly young age group. I'm concerned that reporting AEs may be challenging if it's visual hallucinations, exactly what's going on with our patients who will probably be on steroids as well. So there's that effect in combination. Risk could be greater in the younger population. I think this may slow accrual because if faced with other trials that have drugs, and there are trials with very little to no side effects, it'll be a little bit of a hard sell. And we know, despite the very promising preclinical data, as Malcolm said, the translation to a clinical benefit has yet to be realized. So I think there needs to be thought into exactly what drugs will be used and how this will be addressed, because I see this as a challenge. Thank you.
Yeah, to follow up on your comments, I've had the same concerns about the attribution, right? Given the toxicity profile of this drug and the type of disease that you are targeting, I think that sometimes it will be very challenging to assign an attribution as to whether this was related to progression to the disease or to the drug. So that's going to have to be very well specified in the guidelines of the protocol. I think that Greg raised his hand again. Greg, do you have a comment about the question? I think you're on mute.
Sorry. I think I'm off mute now. Yeah, I just wanted to clarify, Alberto, that the point of this question was not about long-term neurodevelopmental toxicity, but really acute neurotoxicity, risk mitigation, or management. So, you know, we're well aware that in the at least early phases of evaluation of this agent, if the signals of activity are compelling enough, we're not really focused on long-term, but it was really the acute neurotoxicity. And I would agree that particularly in this patient population, attribution of some of these neurotoxicities are going to be difficult to establish. And recognizing visual hallucinations in very young children, I think, will be difficult as well. But that's what we were trying to elucidate. Were really discussions around that issue.
Anybody else has any comments? I don't see any other hands. Okay, so to summarize this, so the purpose of this question was really not to talk about really the long-term effects of these agents, but acute toxicities and how to mitigate the occurrence of these toxicities. Some of the panel suggested that there will be very specific guidelines on how to mitigate the effect, how to make attributions of the drug to the side effects, and the other important thing would also be how to identify side effects in a younger population. I don't know if I've missed anything or if anybody wants to add anything, Toby or Greg or anybody else.
No,
I think you covered it.
I think I covered it.
So if everybody's happy, we will proceed to the third question. The FDA will read the third question.
Hi, this is Denise Casey. The third and final question is, are there non-CNS pediatric cancers that should be considered for evaluation in the Marizomib development program?
If there are no questions or comments concerning the wording of the question, we will now open the questions for discussion. Will?
Was that for me, Alberto? This is Will.
Yes, you raised your hand, yes.
Thank you. Yes, so I've been trying to ask a question previously. We didn't have time. I think this question gets to one challenge of several of the discussions we've had here, which is, based on the previous discussion, I'm actually not quite sure of the specific biologic rationale for which CNS tumors should be studied. I mean, people have asked specific questions about rhabdoid tumors, medulloblastomas, et cetera. There's honestly, as far as I've perceived, not a very specific biologic rationale for why H3K27M, DIPGs, or midline gliomas would be the priority. I understand from practical reasons, or that those are patients who desperately need therapies that we might want to try there. So that actually is what makes, I think, consideration of other both CNS and non-CNS tumor types a little bit difficult. It's also what I believe made discussion of rational combination therapies a little bit difficult, because we could discuss other investigational agents that are relevant to DIPG and high-grade glioma, but trying to make the link to the specific biology of this agent was challenging. So I think, I guess, more a comment than a question, but my comment is essentially that it's challenging for me to to say what other tumor types specifically should be studied beyond other kind of clinical needs or strategic concerns.
Thank you. One of the comments I had is sort of related to what you said, Will, and that was when they presented the data, they said that the CNS tumors outweighed, because of the toxicity profile, some other tumors. So I really cannot think of any other non-CNS pediatric cancers, at least at this stage, in which there would be a very strong rationale for testing this agent, especially with the toxicity profile that has been described and the high CNS penetration. Catherine Bollard is next.
So I guess I was just interested, given that I'm a lymphoma doctor, what the data would be to support a lymphoma indication in a relapse setting for children, given that obviously this was evaluated in adults with lymphoma and myeloma.
I don't have an answer for you. I don't know if anybody in the panel.
No, so it's just putting it out there for Celgene too.
And then we have Steve.
Steve DuBois from Dana-Farber. There is a literature on bortezomib in combination with chemotherapy for children with relapsed ALL that I think showed nice activity, and that's not a primary CNS cancer, but certainly a childhood cancer that can disseminate to the central nervous system. And so I do sort of wonder about that as an option, and would welcome maybe Elizabeth or Andy's thoughts as bona fide leukemia doctors, which I can't claim to be.
Any comments on the leukemia aspect?
Yeah, hi, it's Andy Kolb from, sorry, Elizabeth,
No,
you want to go
please
first?
go ahead, Andy.
Oh, sure. This is Andy Kolb. I represent the COG Myeloid Disease Committee and pediatric oncologist at DuPont Hospital for Children. I think from the AML perspective, we have invested quite a bit in proteasome, studying proteasome inhibitors in pediatrics. I think it would be hard to conduct another trial in AML. There are signals. You could enrich a population looking at signals, but I wouldn't for a signal. But I don't think that would be a primary development path. I'll let Elizabeth comment about T-cell and B-cell ALL.
Thanks, Steve. Just as you, sorry, this is Elizabeth Bates. I think, Steve, just as you've mentioned, it's been there have been some promising results. Bortezomib has been used in a salvage relapse regimen for relapse B and T ALL. And the results from that trial were published and showed some favorable early response rates. And then the ALL 1231 frontline T-ALL trial closed early but had a randomization to bortezomib during induction and delayed intensification for T-cell ALL. And so I think we're still waiting for final definitive conclusions from that study, but I think because it has been, proteasome inhibitors have been studied, I would see that there may not be as much of a path forward there because they've been studied fairly extensively previously.
Thank you very much. Greg?
Thanks, Alberto. And just to address Dr. Bullard's question about lymphomas, I think it's a very difficult space, fortunately, in that the number of children with relapsed non-Hodgkin's lymphoma is really very, very small. And I think at a recent International Accelerate Strategy Forum, proteasome inhibitors were not really high on the list of priority agents to evaluate, given some of the other immune-directed therapies, antibodies, naked antibodies, as well as antibody-drug conjugates, and even possibly engineered cell therapy. So it might be a little difficult. And I think evaluating another proteasome inhibitor in the lymphoid leukemia space. It's also crowded. There are a couple of proteasome inhibitors under evaluation that have the studies have fraught with great difficulty in accruing patients. So you know, the real role for proteasome inhibitors in either salvage therapy or upfront therapy in anything other than T-cell ALL, I think is really a question that is of real interest.
Thank you. Just a friendly reminder that if you've asked the question and to lower your hand. And then we have Leo.
Sorry, I should have put down my hand because Steve asked my question.
Thank you. Are there any other comments on this third question? Will? We have Malcolm and Will. Malcolm, would you like to comment on this question?
Yes, please. That's Malcolm Smith, NCI. A couple of points and some reiterating points made. I think there's a general question of, is there really any place else left to study for proteasome inhibitors in childhood cancers? And there's been a good discussion about AML and other cancers. But I think a tumor that has, an agent that has substantial CNS toxicity really isn't going to rise to the top among the class of proteasome inhibitors. So I think, just from that reason alone, the answer is no. I do want to emphasize Greg's point about especially the non-Hodgkin lymphoma and especially the B-cell non-Hodgkin lymphoma. Greg mentioned the Accelerate meeting, the publication in 2019. I think it listed that there were 13 PIPs already, and thankfully our treatment has gotten so effective for most of these children that the number of children who would be eligible for these trials of novel agents has gotten quite small. And then they're all the T-cell engagers that are exciting to the adult lymphoma docs. They're an ADC that's shown high-level activity and reported at ASCO, looks exciting. The CAR T-cell, there's so many opportunities that I think we really need. The bar is incredibly high for non-Hodgkin lymphoma, and I just think we need to acknowledge that whenever we talk about NHL and new drugs moving into the pediatric NHL area.
Thank you, Malcolm. Dr. Cheng?
Hi. Sorry, I just want to make a comment, so thank you for recognizing John Cheng, industry rep. I think we all want to understand other pediatric tumors, but as everyone recognized, sometimes it's a little bit difficult. I do think it's helpful to distinguish studying other tumors in the context of investigator-initiated hypothesis generating, and then also getting clarity on the context of a written response or written request. And so I think studying it in the context just to understand biology is helpful, but studying in context of a written request, I think should be pretty focused so that it allows the sponsor kind of an opportunity to kind of clarify things. And so as I understand it, Celgene, or the sponsor, has studied a number of tumors and they're focused on GBM appropriately. And so I just want to make sure that that's maybe helpful to understand, maybe from the committee or the FDA's perspective. Those are two different things potentially from a sponsor's perspective.
Thank you very much for that comment. Anybody else has any comments, or I'll try to summarize this very brief discussion on question number 3. So I think the overall consensus was that there's really not, this proteasome inhibitor space in the leukemia lymphoma world is very crowded, and there was not a lot of interest in pursuing this agent in that patient population. There was some concern also about what is the optimal patient population with CNS tumors in which this specific compound needs to be studied. And The other thing that we talked about was what Dr. Cheng just said, that we need to be cognizant of the fact that we need to consider the written request versus investigator-initiated trials if there's a specific biological rationale for this specific agent. I don't know if I left anything out or if anybody wants to add anything to what I just mentioned. If not, Dr. Riemann will now provide our closing comments for the day.
Well, thanks, Alberto. My closing comments will actually be very brief, and I'll start again with a thank you to the members of the advisory committee, and also to our sponsor, Celgene BMS. I think this was an interesting product. I think we had some very interesting, insightful discussions. And again, just want to thank everybody for their participation. Look forward to your participation tomorrow. And again, I just want to apologize for the change in agenda. Since our second day was planned to discuss the relevant molecular target list and look at the specific targets that were or that are the result of specific genetic aberrations and go over that. But given the fact that we had to do this meeting as a virtual meeting, we'll plan for some time in the future when things get back to normal, or when we all get used to the new normal, and hopefully plan a face-to-face open public workshop to discuss the target list. So again, thank you very much for the discussions and presentations today. Thanks.
Thank you very much, Greg. We will now adjourn the meeting for today and continue to the next session tomorrow morning at 10 o'clock in the morning. Dr. Bonner, anything we need to know about is we have the same link to join the meeting tomorrow, the same that you sent this morning, or any other things that we need to know about?
Hi, this is Latoya Bonner. It is the same link, so you'll use the same link for tomorrow morning's meeting starting at 10. And make sure for those who are participating in the actual discussion to phone in 30 minutes before the meeting starts.
Thank you very much, and we will see you all tomorrow. Thank you so much.
Thank you.
Audio recording for this meeting has ended.